person will talk a lot of things. So I would like to say that Dr. Ramchand is from MS Baroda. He did his PhD with the father of our Nobel laureate Venki Ramakrishnan. So he did his PhD with Ramakrishnan, and after that he had a stay in England. Of course, I think he is more English in certain professional things, and he has served as a vice president of research in Sun Pharma, Spark. And then he also did more or less the managing director, vice president of few pharma companies. And but he has in his blood that Gujarati training. So he became more a businessman than anything, but a scientist, a business scientist. So that's great, very rare to see such combination. So he has been creating structures with innovation, because he is a person, I think his appetite does not stop with anything. He was voracious in creating structures, and successful, not just creating and leaving like that, like some people, but very successfully. So he has started a company called Saksin just a few years ago, maybe 6 or 7 years. I remember the start, and it is going very, very successful. And I'm very proud to say that one of the very rare companies who has come up with the CGMP fabrication for the clinical trial within 2 months. So I had the pleasure of being in those, examining those things and finding it extraordinarily good. And not only Saksin, he has created another structure in Chennai itself, where it is a few companies and training, because he has understood the need that the manpower in India is not adequately trained in the right way to serve the industry in the what you call the good science with production. So, and this is what I think one of the very rare opportunities that he has created these structures. And he is also part of many companies like MacGenome, and MacGenome was the one who got the award. So I think I can go on talking, so I don't want to take away his time of talk. So, and also one more thing, he has an infectious disease, which is enthusiasm and energy. So he infects me all the time with that. So I want that he just give it to everybody, and instead of corona, we'll take this as the new thing. So we'll take it from him, that enthusiasm and dynamism and energy. So on to you, Ramchand.
And I'll just share. Good morning, everybody. Good morning, those who are in India, overseas. And my special thanks to Professor Vijay Lakshmi and Professor Krishnan. She was telling about, I have an infectious disease. I felt she is more, because at this age, I don't want to tell her age, but then she's the most energetic person ever I have seen. And, you know, she does not tell no to anything. And that's why, because of her, that great place CBST has come up. And her footstep, Professor Krishnan, also has initiated many things, especially back-to-back 2 conferences, Zoom conferences attended by 100 to 1,000 people all over. It's not an easy thing. So thank you both for giving an opportunity, especially the earlier also I gave a talk on therapeutics as well as diagnostics. Now it's all about drug discovery, development of novel chemical entities, drug repurposing, immune modulators, and reverse pharmacology, which I will be giving. I'll briefly talk about the companies, what we are involved. Mostly it is in Chennai. We have facilities also in Cochin. Now, why I'm going to talk on corona, because, you know, we have some connection with the corona, though coronavirus and its understanding about the proteins have very recent thing, but then certain things we have to do. If you see, the Saksin Life Sciences is a very successful biologics company. We have developed a molecule for macular degeneration, which is in the phase 1 trial, just going into a phase 1 trial. Theragen Biologics is another biologics company. We have developed a molecule for diabetic retinopathy. So both these companies are based on synthetic biology. And synthetic biology is one of the most recent methodology developed for the drug discovery development or biologics discovery and development. And it's very essential for therapeutic development of the vaccines or antibodies or Fab molecule for whether it is coronavirus or similar molecule. MacGenome is other successful company. We commercialized the products a couple of years back, now reached in such a way that, we have break even. And also we got a big grant from BRAC. So we developed few products for corona. One is that viral transfer medium, unique medium. The other thing is that we also got a funding for developing RNA extraction, which we are already commercialized. But then for the purpose of RNA extraction, we are waiting for the approval from ICMR. We are also developing a product from saliva RNA. So other companies are that, Theragen Life Sciences, which is an ANDA company, abbreviated new drug application, where we are planning to do certain repurposing. And the fifth one is SIP-wise, basically functional drinks we are developing. Maybe one or two slides I might be able to show you. And a curcumin-based drink we are introducing, and within few days' time, couple of months' time, an immune booster or immune potentiation drink. I will be briefly talk about that. And another one we are developing is an immunoglobulin vial. So for a throat spray, nasal spray, that is another thing. So we have some connection with the coronavirus currently. And that's why I thought, maybe I might be able to tell about how the drugs or NCEs, biologics are developed. Apart from this, we also have few other companies where I was directly or indirectly involved. We also have a biotechnology, biopharma skill enhancement program, which is funded by Kerala State Industrial Development Corporation. About 10 to 20 students every year we take, mainly training purpose in the various aspects of drug discovery, development, biologics discovery, development, etc. Yeah, sorry, it's moving now. This will be the overview of the talk that I am going to give. I'll briefly talk about drug discovery. And I think yesterday, quite a lot of people have mentioned about what drug discovery, but then I'm going to talk normally not on drug discovery per se, but I'll be briefly talking about how drug discovery can be done for COVID viral proteins. There is a difference is there. The reason is that it's a recent virus. And nobody knows about the protein. So you can't use the classical way of drug discovery over here. Everything has to be very quick. What can be done? The same way biologics, whether it is vaccine or antibody or Fab molecule, how that can be developed. Again, it has to be quick within few months to 1 year or 2 years. Otherwise, there is no fun because the pandemic is increasing and many people that, reaching herd immunity. So unless the drug is developed very quickly, it does not mean anything. Then I'll be talking briefly about the protein targets for control of COVID-19 and what are the targets in host cell as well as the virus protein. Then small molecules under development for COVID-19. Currently, you might have heard about many molecules are coming out. How this so quickly it's coming out? What is the technology they are using? So I'll be briefly talking about that. The same way, novel biologics for targeting COVID-19. Then what exactly drug repurposing? What is reverse pharmacology? What is vaccine development and how it is done? Briefly for the coronavirus. And what is immune potentiation? Please remember, the slides are very complex. So I will try my level best because I know a lot of students are attending. So as simple as possible, I will try to explain. Now, NCEs and biologics discovery and development, what are the major phases? You all know that, the first is the research phase. Research phase is the phase or the discovery phase you can call. That is the time how the target protein is identified. What is the therapeutic area of interest? In the therapeutic area, how the therapeutic protein or the therapeutic target is identified. One of the most complex things ever mankind known is this. Because there are thousands and thousands of diseases are there. Still, we do not know many of the diseases. None of the neurological diseases or something connected with the CNS, central nervous system, brain, nobody knows. For example, schizophrenia, depression, mania, manic depression, Alzheimer's disease. 20% of the people are affected by Alzheimer's disease. Is there any drug? No, nothing, because the mechanism is known. So the research phase is the most important, especially novel target. The second thing comes once the target is identified, you will have to identify what is the drug, comparatively simpler than identifying a target, whether it is biologic molecule or a small molecule. Now, the challenge comes the development of the molecule. Probably it is, for example, if you want to discover a small molecule or a biologic, it's not that great difficulty, because once you have an idea about the structure, then you will be able to synthesize the molecule. Thousands of efficacious molecule you will be able to synthesize. But developing this molecule to reach the commercial phase is very complex. It takes 15 years. That is the challenge. So development is not that easy. Please do understand, only few hundred molecules are there for all the diseases combined together. So the major challenge is that. Then the commercialization phase, that is again, that's the job of the pharmaceutical company. Here, one thing I want to know is tell is that the major research phase is happening world over universities, and companies normally will not spend too much money on identifying novel targets because extremely risky, a lot of money is required. Now, if you see here, this is the drug discovery is complex and time consuming. Now, I gave this over here just to let you know, normally it's about 12 to 15 years and $2 billion is required for developing a drug, whether it's a small molecule for COVID viral targets or as a vaccine or an antibody. So if 15 years takes a drug to develop after conceptualizing, understanding the target, what is the fun in developing a drug for COVID? So you can't go through the classical way of drug discovery process. If you really see, there are multiple phases out there. There's a strategic research phase to understand about the target, what type of protein you should select, and how to identify the target and to validate the target. Then there is an exploratory phase in order to generate the hit molecule, then hit molecule to lead identification. Multiple things are done, whether it is virtual screening or assay development or high throughput screening. Then drug to redesign to get the best efficacy and safety, and finally the hit to lead molecule. Then lead optimization, and animal models is very, very important because most diseases do not have an animal model. Unless you have an animal model, you will not be able to tell eventually this is going to work or not. And that in the animal models also you can do not only efficacy, safety, but the absorption and its metabolism, excretion, distribution, etc. Then once the molecule enter into the clinical trial, it takes another 6 to 7 years. But let me tell you, all small molecule, 80% fail during this time. And during 10,000 or 5 to 10,000 molecules, 1 or 2 molecules enter into the clinical phase. I just wanted to tell you the complexity in drug discovery and development, then why we should go and do a drug discovery process for a pandemic like coronavirus. That's what I want to tell. So the classical way cannot be done. Now, I'll also mention about, drugs and biologics discovery and development. What are the different types of chemical or biologics entities available? Let's say that, let's go through that. If you talk about biologics, there is novel biologics. If the target is absolutely novel and your antibody or the Fab molecule or the CDR is novel, that is called novel. And another one is novel biologics. or it can be also called biobetter, in which the target and mechanism is same, but CDR, complementarity determining region. This is the paratope which goes and binds to the epitope, or rather the antibody goes and binds to the antigen. That will be different. And there is something called biosimilars. India, it is very popular because whatever is the novel drugs, novel biologics developed in US, Europe, we develop it as a biosimilar. But biosimilar development also is not that simple. Exactly similar pattern of the sequences, its metabolism, configuration, conformation of the protein has to be there. Now comes small molecules. Please remember that not many NCEs are developed in the world, only 420, and there is another category called Me Too drugs based on the existing NCE, a structural analogs. These are called Me Too drugs. So India has not developed the NCE, only 7 countries in the world could. So NCE is another one. ANDA, abbreviated new drug application, which is mainly that drugs or generic drugs that is to be introduced European US market, that has to go through a very different process, comparatively difficult, takes about 3 to 4 years or 2 to 3 years. Then comes generic molecule. Most of the pharma, Indian 26,000 pharmaceutical companies are there. Out of that, 99% of the companies are doing generic molecule. What is new chemical entities? You can just see a protein and to which a molecule goes and binds. I have shown some simple structure in the left-hand side and how this goes and binds. A molecule with a novel structure that is newly identified as potential drug, and that is going to be the key ingredient. When you are developing a drug, the structure, there is some requirements are there for the structural aspects of the molecule. You can't develop the pharmacophore for anything and everything because the molecule should have very good bioavailability. It should go around the body, enter into the cell, only go and bind to the target protein. So it's a very complex process, and the interaction normally with the target protein and your drug is multiple. It is hydrophobic or hydrophilic, electrostatic, hydrogen bonding, van der Waals forces. So multiple forces are involved in, and this binding has to be reversible. Now, designing NCEs and biologics is very complex also. It's not very simple because the specificity is very important. You can't go the drug or the biologics to go and bind here, there, then it will develop toxicity. We have to be very careful that the molecule should be absorbed or administered properly. It can be an injectable or it can be given orally. Then, it should be distributed throughout the body. There is a binding process to the body proteins are there. That is the way it is distributed. It should be appropriately metabolized and excreted. Otherwise, it will become toxic. It's very important that, to reduce the adverse effect and improve efficacy. And it's also to understand the right dose, right drug, and the right time. What time to be given, once in a day or 4 times a day. These type of things has to be there. So these are, and it is also important, FDA or no regulatory people will approve without understanding the mechanism. Because what is the drug structure, where it goes and binds, what is the transduction process, everything should be. So it takes several years. And it is challenging too. Especially the development process is extremely challenging. What is challenging means, the uncertainty of development. If you are developing a biologics, Professor Vijay knows that, the complexity, bioseparation, the molecule should be separated with all the other toxic substances or other proteins. Scaling up, because from lab, like 100 ml or 200 ml to 10 liter to 100 liter, finally to 2,000 or 5,000 or 10,000 liter, lot of problems you can face. Analytical challenges. For example, if you are developing a protein, there is at least 20 different analytical tests is there. One is its configuration and multiple conformation, the formulation challenge, then the stability of the molecule, then the multiple assays, surface plasmon resonance, if you are talking about an antibody, or the the antibodies affinity. So all this has to be that, these are the challenges during the development process, and all has to be sorted out. That's why it is extremely difficult to develop, whether it is biologics or an NCE. That's why very, very few companies could develop that one. Let me also tell you that we are the only single company who has gone through all the RCGM requirement to get into a phase 1. One of our molecule is reached the phase 1 trial in totally novel biologics, which is against VEGF, vascular endothelial growth factor, for macular degeneration. And it's a very time-consuming complex. Now let us see about the viral, how we can develop drugs against virus. Please remember, if you are talking about the human body, all the targets we know very well, and it has been last 100 years or 70, 80 years, this has been going on. Now, many protein structures, configuration, conformation, three-dimensional conformation, its transduction process, everything is established. So what we need to do is that develop the drugs against that. But then that is not the case with the virus. Suddenly a virus has come and the pandemic has started. You can't develop a drug against that. So what is to be done? Now the scientist has a methodology immediately to sequence and predict the structures, and you can go through X-ray crystallographic structures, and that is what is done. So there are methodology to understand the structures, these novel structures of the protein. That has been done. Now, if you see about this virus, what the virus can't replicate by itself. It needs other organisms like a bacteria or a host cell is required. Now, these protein, there are not too many proteins. If you are talking about the human body, which we have about, we have about 25,000-30,000 genes and probably so many numbers of protein. But in the case of virus, probably 60 or 70 RNA is there. Probably it produces 30 or 40 proteins, and many of the proteins are inside. Why I'm telling these proteins are inside, it's not easy for a small molecule to go inside and work against the protein. So it's not possible. So you will have to find out what are the proteins which is available outside so that the molecule can go. And few proteases are there. So mainly these virus contains some of the functional protein. There is structural proteins are there surrounding that, but it is not easy to target a structural protein. The reason is that structural proteins do not have a binding site to alter its function. But if you see about proteases, it has got very clear-cut functions are there. There is papain-like proteases are there. Then there is other few proteases are there. Then there is RNA-dependent RNA polymerase. These are some of the 3 or 4 very good targets which has been identified that can be used as a drug target. Now I have also given that antiviral activity targeting the viral metabolic function, which these 2 proteins, proteases and RNA-dependent RNA polymerase is involved in the metabolic function. The other thing is that you can also target the host cellular function. Why host cellular function? Because as I mentioned, the virus has to interact with the host cell. One is that, it interacts by going and, interacting with some of the surface protein. In the case of coronavirus, there is a enzyme called angiotensin converting enzyme, which acts as a receptor. And there is a spike protein surrounding the virus. That protein goes and binds to this angiotensin converting enzyme. And after binding, it goes inside. A fusion takes place, goes inside. Once it goes inside only, the viral replication can take place. So if you inhibit that process, it's possible. And the target host cellular function of that particular protein, angiotensin converting enzyme, already few drugs are also there. So how this can be used? The same way RNA-dependent RNA polymerase that is involved in the development of certain other proteins within the host cell, and how that can be inhibited and the virus can be killed. So these are the things what the scientists are working on. So this is basically some of the protein targets that I have mentioned. If you see what you are seeing above, this is the coronavirus, and these are what is spike protein. And you can see one is the spike protein interacts with the angiotensin converting enzyme, and once it do not interact, it cannot go inside. So this interacts, and one is this particular interaction allows to fuse with the cell and goes inside. Okay. Now I have also given a drug that Ramipril, which is basically, I think it is a hepatitis C. Sorry, it is a blood pressure. It is involved in the inhibiting angiotensinogen to angiotensin. So it's a blood pressure drug. Now, this has been shown to bind to angiotensin converting enzyme. If this converts, if it inhibits, then this interaction will not be there. So I think Ramipril is already a repurposed drug. Now, if you see, I've also given some other protein. This is a host cell. It's a protease. This protease is involved with the activating this angiotensin converting enzyme. So this means that, unless this protein is there, you will not be able to activate this angiotensin converting enzyme. So there is already a drug called the chemostat, which is also currently being developed or under development to inhibit this particular protein. And once this particular protein is inhibited, the virus will not interact with the angiotensin converting enzyme. So this is another repurposed drug. The third is CD147. CD147 is another drug, another surface protein, which is otherwise mainly involved in the purpose of, CD147 is mainly involved in the purpose of certain diabetic complications, and another one is connected to, I think, asthma. So there are few drugs are available, mainly antibodies. I think it is under development. So as I mentioned, the diabetes and asthma, and there is another molecule called furin. So there are few molecules is under development. So I just wanted to tell you one thing, that both the virus as well as the host protein, there are certain targets which is already identified. The molecules are there. This means that you don't have to go through the very complex process of discovery and development. That means probably 3 months to 6 months time, the drug can be introduced, because many of these drugs are already tested in human. This is what is called the repurposing. It's very, very important such methodology to be used, because you can't wait for such a long time. And what you are seeing is that multiple protein, whether it is spike protein or membrane protein, envelope protein or nucleoprotein, probably can be used as a target for the development of vaccine. Mainly spike protein is used as vaccine, but many of these proteins can't be used as a small molecular drug, because these are all structural proteins. Now, ideal protein targets in SARS-CoV-2 viron, one of the molecule is a spike protein. Okay. Now, spike protein, as I mentioned, basically it is involved in interacting with the angiotensin converting enzyme. So if you see here, there is a good interaction. What you are seeing here is the complete, above this, the complete RNA sequence details, which is about 70 different RNAs are there. And down you can see multiple proteins, and some of these proteins are certain functional proteins, like papain-like protease. Then another protease, which is 3CL protease. Then RNA-dependent RNA polymerase, and certain endoribonuclease. So these are some of the proteins which has already been used to repurpose existing drugs for that. But S protein, I think mainly can be used for the purpose of developing antibodies or vaccines too. Now here, this is an interesting slide which shows various drugs developed or under development that can be used for targeting coronavirus. As I mentioned, there are three phases are there for the virus. One is the virus has to fuse. Then in order to fuse with the cellular membrane, go inside, it needs another target within the host protein, which is angiotensin converting enzyme. Once it binds to angiotensin converting, it fuses, goes inside. So as I mentioned, there are few drugs that can inhibit this process. Second one is endocytotic process. Now, endocytosis can be inhibited by chloropromazine. Very, very interesting. I was thinking about this particular opportunity with chloropromazine. I just went through the literature. People are already working on that. Chloropromazine, as you all know, is one of the most important antipsychotic drugs, but it was never developed as an antipsychotic. It was originally about 70 years back developed for the nausea and vomiting in pregnant women, the very first one, the GABA receptor. Subsequently, it was repurposed for anti-H1, for that is mainly used for allergic purpose. Subsequently, this has become an antipsychotic drug, which is dopamine D2 receptor. Subsequently, in 15 years back, it has been used for diarrhea purpose. That means it has been shown to inhibit the cyclic AMP, the production of adenylate cyclase. So inhibit adenylate cyclase. Now, this has already known chloropromazine has a membrane stabilizing ability because of its hydrophobic, hydrophilic, as well as charges. So now this is has reached the phase 3 trial, chloropromazine, for inhibiting the endocytotic process of coronavirus. So you can see over here multiple ways by which a certain existing drugs can inhibit. So one is the transport of the virus inside the cell. After reaching that one, there are 3 host cell proteins are there, already existing drugs. Everybody heard about chloroquine, how that inhibits, but I'm not sure that how effective that one. But scientifically, that structurally it is possible. But then it is very, very important. Unless this is proven in a clinical setting, it does not mean anything. This is the unfortunate thing, that we can, scientifically, in vitro, lot of things can be brought about, but it has to be proved in a clinical setting. But let me tell you, I don't think there are at least 5 or 6 drugs are there which is not very, very clearly proven that this can be used for coronavirus, though people are using that one. So maybe after a few years or a couple of months, we will know very clearly whether really it is effective or not. So chloroquine, then chloropromazine, then tetramidine, another drug. Then there are few other drugs are also in the process of development over here. Now, the second is once it goes inside, what is happening is that the virus will have to get involved with the host cell, the DNA. Then the replication takes place, which is the main enzyme is RNA-dependent RNA polymerase, and there will be a genomic RNA will be produced for the replication purpose, and which produces subgenomic RNA, which is translated into proteins, combined together, the virus will come out. This virus will be later used for the production of the adaptive immunity within the host cell or within the animal. So this is basically the thing. What I'm just showing is that how the virus is transported inside. After entering that, what all things can be done. Let me tell you, all these drugs currently being tested or already introduced, but remember, everything took only 3 to 4 months time compared to 15, 16 years time. This is the power of repurposing the existing knowledge we are using, because at this point, nobody can think about developing a drug and, and test it. By that time, the pandemic will come, herd immunity will come, everything will become useless. So this is another molecule currently being developed. It is a nucleoside, ribonucleoside analog. So you can't introduce a ribonucleoside itself, because always, if you want to a stable molecule within the body, it has to be analog. So this is an analog of ribonucleoside, and it attacks RNA-dependent RNA polymerase under development, but which is a new molecule. So people will know very soon that how this is going to be effective. Now, if you see over here, I have mentioned about 3 different proteases, which is coronavirus proteases that is being tested for various drugs. And probably this might be using, I'm not sure whether indinavir, which is an HIV protease inhibitor, then ketoamide, and another one is GRL0617, probably under development. So within few months time, it might be used. But if it's a new synthetic drug, it's not going to come very soon, because you have to go through all the phases of drug discovery. If it's an existing drug, it is repurposed. Maximum time required is 2 to 3 months time. Now, what is novel biologics? Now, that's what we are doing. Novel biologics is, it can be an antibody, a full antibody, part of the antibody, Fab molecule, single chain molecule, or the molecules can be combined together. The Fab molecule can be combined together to interact with multiple targets. Now, classically, I have shown here that, that the antigens are injected into animal, and there is a process to develop the antibody. It was not very successful because you can't produce very effective molecules this way. Though earlier some vaccines can be done successful, but no antibodies can be developed. But things have changed. It's by synthetic way. One is using a technology called phage display technology can be. That's what we are using. Subsequently, all this DNA can be synthesized. DNA can be synthesized, can be introduced in the plasmid, then, transcription, translation, and your protein can be generated within the host cell, bacteria, mammalian cells, and everything. So by using this synthetic biology, you can now synthesize anything, including a cell. You will be able to develop basically a cell. For example, membrane, if you want lipid can be synthesized the same way, every DNA, every RNA, every protein. So that's why it is called synthetic biology. Synthetic biology is coming up in a very big way, but currently extensively use the synthesis of DNA for the recombinant DNA technology, and nobody is going to use the classical way of producing antibodies. But there is a very clear-cut difference is there between when you are developing a small molecule or a large molecule. Small molecule is, certain aspects is very complex in small molecule development because thousands and thousands of molecules has to be synthesized. Because it is coming from outside, synthetic molecule, you can never predict about its toxicity and pharmacokinetic profile. And it can give toxicity because it can go and bind to many other molecules. So on the other hand, biologic molecule is not like that. Complex in developing, especially that in order to get appropriate yield successfully for commercially successful product to develop. So major differences are, low molecular weight, large molecular weight, or usually organic or chemical synthesis made with recombinant DNA technology. Few critical process steps, but many critical process steps and in biologics, well characterized, less well easily characterized because of the conformation of the protein. Configuration is well characterized, but then what are the types of bonding? What type of hydrogen bonding interactions? It's very difficult to predict and develop. Non-structured structures may or may not be completely defined or not. Homologous drug substance, homogeneous drug substance, heterogeneous mixtures. Many of the molecules can be heterogeneous, especially when you are talking about certain vaccines and other things. Normally, small molecules are not immunogenic, but this can be immunogenic, especially highly humanized molecule when you are giving to animals. This can be immunogenic. So otherwise, if it is less, not fully humanized, then when you are giving to humans, it will be a problem. It can be immunogenic. I briefly mentioned about synthetic biology. So if you see the left-hand side, you can break down an antibody and make it to the multiple Fab structures or constant region. But that is no more done now. It is all done by synthetic biology, and you can link, for example, two Fab molecules like this, and one can target one CDRs can target to one target. This can go into another one. Now, if it is a trifunctional antibody, if you want to, for some reason, that is possible. A single chain can be made. So any different types of antibody fractions, small molecule. So there is significant increase and improvements has happened in synthetic biology. You might be able to see a lot of interesting things in the future. So this is a molecule that we have, involved in developing. If you see, this is an Avastin, full 180 kDa molecular weight. You can see here light chain, heavy chain. This is a constant region. What we have done is that this part we have synthetically developed. So if you see over here, so this is basically interaction of a Fab protein of Avastin to VEGF, but this is our molecule. Sorry. Yeah. So what you are seeing is that, this is a dimer of vascular endothelial growth factor, which is a dimer. And these molecules are Fab molecule, and how it is connected to VEGF and how it is neutralizing VEGF. That is what it shows. VEGF dimer binds to, for example, ranibizumab. Ranibizumab is Lucentis. I'll just briefly discuss about what exactly is drug repurposing. I briefly talked to you what is repurposing earlier. A typical example is chloropromazine. Now, if you see the chloropromazine, all the evidence has come from the clinical or during the clinical testing, clinical evaluation. As I mentioned, chloropromazine from the beginning, 70 years back, now it is used for corona purpose. So 5 different applications are there. Now, for example, 15, 20 years back, if you have mentioned that, this drug goes and binds to 15 or 10 different receptors, probably it would not have been, FDA would not have been granted the approval, probably assuming that this might develop some toxicity. There can be problem. But anyway, it does not have toxicity, and it is a repurposed drug. So normally repurposed drugs include approved, investigational, discontinued, or shelved therapeutics. So you can use an existing drug, which is off-patent drugs. Sometimes it is investigational. There is a commercial requirements are there to do that one. Many times discontinued drugs. What will happen is that the drugs during the efficacy trial might fail. It would have never reached the trial if you are showing toxicity or problems with the pharmacokinetics. So a drug without a problem with the pharmacokinetics, low toxicity can be used for the repurposing. There are a lot of shelved therapeutics are there. So an universal strategy for neglected disease. What is neglected disease? There are, as I mentioned, 35,000 human diseases, about 25-30% is neglected disease. Neglected disease in the US FDA terms, people suffering from 200,000 or less. For example, this is a pandemic, it can't be a neglected disease. Schizophrenia is not a neglected disease. Diabetes is not a neglected disease. But diseases like Tourette syndrome, very rarely happening, a neurological syndrome. So that's a neglected disease. So in neglected disease, repurposing is a very good strategy. Or diseases like coronavirus infection pandemic, which you have to develop a drug very quickly. So what are the ways by which is done? Pre-existing drugs, where there is a clinical evidence is there, very good clinical evidence, FDA approved with the similar structures, or FDA approved drugs with side effects can be used. Then failed drugs, but failed in terms of efficacy, not by toxicity. Of course, we can't go ahead with a drug with toxicity at all. Now, what are the concerns when considering a drug for repurposing? Is it off patent? Because if it is still under patent, you need to negotiate with the company which owns the patent. Is it FDA approved? If it is an FDA approved, your whole process will become very easy. Is there prior art for it being used in new indication being studied? Most important thing is that if you want study and original patent, the medical application has been, already been covered, then you won't be able to do. You will have to negotiate with the company. Is it still being made? It's very, very important that you know whether the drug is available. Otherwise, you will have to start synthesizing the molecule. Does it work in a relevant animal model for that? This is being studied. It's important that if you are going to understand about the new mechanism, then is it possible to, or the animal models are available. Does it work better in combination with another repurposed drug? That is also can be done. So two molecules can combine together. But people are using a lot of collaborative effort to develop molecule. Let me tell you, it's an extremely good thing, repurposing of drug, good science, good bioinformatics scientists is there. Because all these complications of 15 years, but the value can be generated quickly. But you know, a very good scientist, a teamwork is required developing repurposed drug. This is a very typical example of repurposing of existing drug for coronavirus. Left side, you are seeing a target, which is a protease, I think. The right side, and another RNA-dependent RNA polymerase. Left side, hepatitis C drug, remdesivir. And you know that that is very much used now. Now, what was originally remdesivir is used? Hepatitis C. I think it is last 15, 20 years it is being used for hepatitis C. But now suddenly, the scientists have found that, you know, this particular molecule can bind to a protein which is present in coronavirus, which has got homology with hepatitis C, that particular target. That is the way they came across. And after screening, they identified that one. The second thing is that an anti-HIV drug, lopinavir. So lopinavir is mainly RNA-dependent RNA polymerase. I think that is being tested or introduced in the market already. Now, drug repurposing does not take too much time. As I mentioned, if you have evidence, one is clinical evidence. The second possibility is that you can screen using docking studies, because most of the protein has got X-ray crystallographic structure. All details are there. In vitro or in silico, you will be able to screen and develop novel structures. But the drug is already tested, no toxicity, and clinically available. You don't even need to do a phase 1 trial. Now, normal time, they will ask for a phase 2 and a phase 3 trial. But at this time, if you are developing against coronavirus, probably immediate approval can be there, so that, you know, a small phase 3 can be done, and then they will introduce in the market. And so that, you know, if it is going to be useful, is a great thing. So normal repurposed drugs is about 2 years against about 10 to 17 years for the normal drug discovery program. But the current phase of this pandemic, many of these drugs are getting approval in 1 to 3 months time. Straight away they are giving approval, but they know the dosage is not tested at this time. The dosage they are using, the existing dosage. So that's one important thing, because it all will take time. If you want to establish the right dose, for example, remdesivir, I don't think they are doing a new the dosage for that. Probably currently using is that existing dose only. So I'm just in a cartoon format, just mentioning how drug repurposing works. It is normal drug discovery, very complex, goes through all the processes. On the other hand, in repurposing, especially clinical evidence and, you know, existing drugs, it is very quick, probably few months to 1 or 2 years time. Now here you can see currently some of the approved drugs. I mentioned what are the targets, whether it is mainly involved in binding to the host cell protein or the transcription translation after going inside and, you know, combining together, coming out. So any of these targets, whether it is viral target or host cell target, can be involved. So there is nymphomastat and chemostat. Mainly involved in, the first one is a protease inhibitors. The second one, chloroquine, is angiotensin converting enzyme. The other one is chloroquine and hydroxychloroquine, that is also involved in endosomal acidification process. Remdesivir and flavipiravir, which I already mentioned, which is RNA-dependent RNA polymerase. Lopinavir and ritonavir and disulfan. So these are also proteolytic enzyme inhibitors. There are a few more drugs under development, cyclosporin, which is an otherwise immunosuppressant. So what I just wanted to tell is that there is few repurposed drugs currently available. The repurposing is comparatively easier, quicker. Within a couple of months' time, people will be able to develop. But you need to go through the clinical evidences, and also the structure of the protein of the virus or host cell is very, very important in order to prove and get into the clinical trial. So this is basically the mechanism of remdesivir, how this is happening. For example, after seeing this, I think that a combination of angiotensin converting enzyme inhibitor along with remdesivir could be a good opportunity. By the way, remdesivir is a prodrug. That means once it goes inside, part of it is broken down and become active drugs. You can see the right-hand side is the active part. This is the prodrug. There is a hydrolysis takes place here. This ester bond is hydrolyzed and you get this molecule. Once this goes inside, the translation of the viral polymerase RDRP is taking place. Then this is exactly what is happening within the cell, host cell. So a genomic replication and the RNA is produced, which produces subgenomic or transcription process. Many of these proteins are formed. Now all these things combined together, the virus is formed within the host cell. This comes out. Then this is getting involved in its replication again, and also during this process, this will be attacked by macrophages, antibody will be done. Now, the question is that if your antibody is the antibody production is immediate, very quick, if you have an extremely high immunity, you don't have to worry. If that takes time or your innate immunity is not taking care, there will be a problem. A few more slides. I will very quickly finish. I know the time is getting very closer. What is reverse pharmacology? Another approach, something similar to repurposing. Reverse pharmacology is that in which the classical way is that, right from target identification to lead optimization to preclinical studies and clinical trial, which takes several years. Now, if you have an evidence, and normally reverse pharmacology works well with classical phytochemical or the other Ayurvedic type products, it can be used. But the first thing is that the clinical evidence is required and you should be able to enter into the clinical trial. Now, if a new molecule is there, it takes years and years to get into clinical trial. Already, suppose you have other evidences are there in the clinical evaluation, then you can test for other applications. Yesterday somebody was mentioning metformin is a classical example of such a thing, that in which metformin is originally used for diabetes and few other things. Now it is used for certain cancers like breast cancer. You all know about another very good application is this drug which is involved in inhibiting platelet aggregation, which is aspirin. Aspirin is another a repurposed and using reverse pharmacology approach, which has been done. So what I want to tell is that, reverse pharmacology approach is an extremely good technology where if you have certain evidence and you can go into clinic, then you can study the pharmacological part, then where it binds, what is the mechanism, all can be established, and probably 1 to 3 years time the molecule can be done. This is a very good method for developing drugs or at least active ingredients from Ayurvedic or plant-based products. See, plant extracts, if you want to test in human, as long as it is not purified, you can do under very easily. If it is, as long as an extract, if it is being used, then you can study whether it is in animals or humans, which disease it can control. If it really controls certain disease, then you can study further that extract, purify the product, and develop it as a drug. So just I wanted to tell that, reverse pharmacology is an excellent method. So I'll just briefly tell about the vaccine development. All of you know about that one. What is innate immunity and adaptive immunity? Innate immunity is very important. I just wanted to tell what is immune potentiation. Really clear evidence is there or not. I wanted to tell that. In innate immunity, you know about physical barriers. For example, your skin is a physical barrier. Your nostrils, there is some epithelial, layers are there. If you go to your stomach, there is acid is there. These are all physical barriers. Now, what we are trying in this time, the most successful method is the physical barriers. Currently, we are using, either we are using a mask. If it is not the mask, social distancing, or washing the hands continuously so that organism is there. Everyone knows that this is a very successful method. There are chemical barriers are also there. Chemical barriers, for example, in your saliva, there is a proteolytic enzymes are there. For example, if it goes into the mouth, that proteolytic enzyme or lysozyme. For example, in the tears, lysozymes are there. If an organism goes, virus goes, that can kill the virus. So these are all very good methods. But activating this or increase this activity, I don't think anyone knows. Then once this virus somewhere or other goes through our physical barriers, goes inside, we have many other methods by which within the cell, within the few hours, you can see. These are NK cells are there, dendritic cells are there, basophils, eosinophils, neutrophils. These leukocytes can immediately kill by multiple methodology. So now, innate immunity, if you have an extremely good innate immunity, that will protect you. Now, who has very good innate immunity and who do not have, how to activate that one? That's a billion-dollar question. Because people, you will be able to see hundreds and thousands of products. People are telling to take vitamin. People are telling to take minerals. People are telling some Ayurvedic product. People are telling exercise is good. Some people are telling that, exercise with your stomach. Some other people are telling pranaya is good. Or, your the respiratory control. So many people are telling, and whether you are doing swimming or running or jogging or walking. So many things. Some people are telling very good nutrition, but let me tell you, nobody has proved that which actually, because if it is to be proved, it's not in vitro. In vitro, many things can be proved. In a clinical setting, under controlled condition, blinded condition, this is to be proved. For example, someone is telling you do this one, your natural killer cells can increase significantly 20%, 30%, then it is understandable. Until that time comes, or someone is telling that ginseng can increase so much. In vitro is fine. I worked on curcumin. Curcumin has got 11,000 applications, probably almost 45. I myself have got about 30 to 40 patents are there. One of the first highly bioavailable curcumin is introduced in the market, but not yet proven clinically. Unless that is proved clinically, it does not mean anything. Now, adaptive immunity, there are two ways.
Ramchand, I think you have to wind up. Yeah.
Okay. Yeah. So probably, I think then, can I have 2-3 minutes?
2 minutes, 2 minutes, because it's very interesting, extremely interesting. But unfortunately, time is a constraint. Yeah,
Okay.
go ahead.
Yeah. In adaptive immunity, you have your helper T cells, cytotoxic cells, that will few days it will work. But the major thing is that if you are attacked by the virus, then your immune cells will take over, then antibody will be produced. There is a memory cell as well as, plasma cells. Plasma cell will secrete your antibody. Your antigen or the virus will be immediately neutralized. Then you get an attack later. Then your plasma cells will memory cells will release the antibody and you will be safe. So I'm not going to go ahead with any of my slides. Just I want to tell that many of the vaccines are under development, whether it is attenuated or inactivated or weakened virus can be used. Viral vectors, it is being successful in animal vaccines, not yet in humans. The same way nucleic acid is under development. Most successful is that protein-based, whether it is protein subunit or virus-like particles. So inactivated and weakened, very successful. Protein-based, very successful, currently under development, whether viral vector or non-replicant. So these are the different types of virus under development. So vaccines, I know how it is developed. If you have a weakened virus or inactivated virus, it can be introduced. Then what will happen is that your immune system will take over by dendritic cell or macrophages that will interact with T cell, B cell, produce your immune antibodies, and that can kill your virus. So these are some of the major vaccine in development. I just already mentioned about what is immune potentiation. Now, immune potentiation is that whether is there any real drug or a supplement really to potentiate, increase your immunity. There are hundreds of products are there in the market. Constantly it is coming up. You know that, in the airport now everybody is taking curcumin-based drinks. Is there a proof? If there is a proof, you need to have clear clinical evidence. Until that time, there is nothing wrong in taking. We will have to assume that it increases your this thing. For a scientist to believe, you need to have real clinical evidence. So I'm not going anymore. This I have also given a slide on ginseng. So many people world over is using, but most important thing is that these are all in vitro evidences, but clinical evidence is required in a clinical setting. So, and we are developing another antibody-based therapy where it is called immunoglobulin Y. That's actually in collaboration with a US company. We are in the discussion process. You can produce immunoglobulin called IgY, or in the yolk it will be produced. Then this egg yolk can be used, your antibody source, and a very interesting technology. And we are trying to develop as throat spray, nose spray, a drink, etc. So I think, thank you very much. And yeah, thank you. Thank you very much.
Thank you. Thank you, Dr. Ramchand. I think extremely rich. I don't know. You are very good in making companies and financially strong and also scientifically too rich. So I think I have a very quick question before I give it to the participant. See, in the drug repurposing, when you compare small molecules versus protein molecules, how would you say in terms of time limit and the usefulness, which will be the, I think, obviously it must be small molecule, but I want your comments on that.
Yeah, see, normally repurpose is very successful in small molecule. The problem with large molecule, fab molecule,
I was going to say
that it is highly specific, because CDRs are highly specific.
And also the 3-element structure will just go and bind somewhere. That was the purpose I was asking.
Correct.
The question.
With the same target, different application is fine. Something similar to that. For example, let's say in our case, VEGF is involved in multiple.
Yeah, I know. Yeah.
So wherever that can be done, one is it is involved in any angiogenesis process. Anywhere angiogenesis is used, it can be used. So that is one way of doing, and it can be used, biologics can be used. But the real repurpose is that small molecule, multiple targets, identifying multiple targets without any toxicity.
Thank you. So I think maybe some questions.
I will request Dr. Sabris to take a few questions from participant.
Yeah,
Yeah.
thank you, Sanjit. A few participants have asked questions. One participant is asking, in drug repurposing, how do we decide the dose of the old drug
That's
for
what he was
new
saying.
activity?
Excellent.
That is what he was telling.
Yes, which is I mentioned that one. See, currently under this condition, because if you need to identify the right dose, it will take time because we need to evaluate. A typical example, as I mentioned, is aspirin. Aspirin is a COX inhibitor, which is also a leukotriene inhibitor for platelet aggregation. In the first case, it is 200 to 400 milligram. In the second case, it is 50 milligram. It takes some time to establish the dose. So, but there is nothing wrong that, at 400 milligram also, it can be a platelet aggregation inhibitor, but it is too much. So, but, in an emergency, you can use. So what is companies are doing in an emergency, existing dose is used, but during the process, they will be able to come out with the right
Yeah,
dose.
but there is one problem. If the existing dose is not efficient, then you have to increase. So that where we'll have a hurdle.
Right. If the dose is, for example, I had exactly a similar thing for hepatitis C when we were developing. The dose was about 3 times. The Indian regulatory did not give us approval to go for hepatitis C.
That's a very important problem. Yeah.
They told we will have to do a dose response phase 1 trial. It is as good as any other trial. But then we did that in Belgium. This is for chemists.
Okay. Thank you, Dr. Ramchand. Another question from the participant is, can adaptive immune response work if a person is reinfected with COVID? If it works, then what is the lifespan of antibodies generated?
Right. Now, it's again an interesting question, but the thing is that this reinfection is not proved. Let me tell you, the reason is that if the virus is infected, normally everyone knows that it will be there for 7 days. After that, the antibody will be formed. But see, what you are testing, either an antigen testing or you are doing RT-PCR. Now, viral particles will be there. Now, viral particles has been shown to be, or dead virus is shown to be present up to 3 months time. Now, what you might be testing in RT-PCR or antigen testing is probably an existing one. Nobody has proved after 2 or 3 months time live virus is present or not. So I doubt about that reinfection that is to be proved probably, unless the virus has got mutations takes place. You are coming out with a new protein within the same virus, then it's a different thing.
Yeah, but there is also one possibility which has been shown in other virus. Even if the reinfection happens, it will be mild. It will not be very severe and tough.
Correct.
Yeah.
Okay. Do we have time to ask one more question, Sanjit?
No,
Quickly.
Sabrish. Okay, take one question, Sabrish, and then.
Okay, this is the
we'll
last
go to
question.
Next speaker.
Yeah, okay, one last question. Azithromycin is known to inhibit the spike protein CD147 interaction. But clinically, hydroxychloroquine azithromycin combination was not effective. Can we assume meplazumab, that is anti-CD147 antibodies, won't be effective as well?
Right. I have mentioned about 147. That is basically that antibody is for the purpose of diabetes and asthma. I'm not sure whether it has come out or not. Probably it might be useful. Probably, because if you remember in one of the slides I have mentioned about the CD147 antibody, how that is involved in the interaction with the corona's entry into the host cell.
Right. So I hope the participant name is Madhu, whose question is answered. Thank you, Dr. Ramchand, for a very informative and very nice talk.
Thank you, Dr. Ramchand. So thank you again. I think we will have certainly a lot of interaction and share our
Thank you, Professor
efforts. Yeah.
Professor Sabris, and especially to Professor Krishnan. Thanks a lot for this opportunity. Thank you very much.
Thanks very much.
I will stop my sharing of the slide.
Yes.
So can I start?
Yes, ma'am. You can start, ma'am. You can start.
So now we have the pleasure of listening to Dr. Ramesh Warrior. So Dr. Ramesh Warrior is one of the icons in India for Ayurvedic treatment. I'll come to the thing why I say icon. He is not the first generation. He is the third or fourth generation in that, continuing the efficiency and continuing the confidence of the people, because I have been myself a patient there. So Dr. Ramesh Warrior, he is he is the managing director of Arya Vaidya Nilayam Arogya, that a new name, yeah, because I know only as Arya Vaidya Nilayam. And Arya Vaidya Nilayam, which was existing in Madurai in the beginning, are also involved in manufacturing the Ayurvedic productions with standard quality. I remember almost 25 years ago, they were having a possibility of analyzing and then standardizing the preparations. I think that was the very good beginning of the effort to see how they can really guarantee the quality. And also they have the now Ramesh Warrior, after his basic degree in Ayurvedic medicine, has done MD, and also he has done the management to understand how to really bring up the structure, which is now a very, very important structure in India. Not only in India, and I think he has crossed the borders, because I know that he has been into different European countries and setting up the facility as well as teaching them what is exactly the Ayurvedic treatment. So I think this is what is needed today, because Ayurveda is a very old system of thousands of years. But people are being kind of secluded, Ayurvedic people and the modern people. So that seclusion has created a lot of difficulties and doubts and non-standard product coming in the name of anything. So I think Ramesh Warrior's effort, the pioneering effort in that is very important. Continuing that effort, he has set up very recently, I think two or 3 years ago, a small research R&D sector. R&D sector trying to bridge the knowledge gap in the Ayurvedic medicine, in understanding using modern biology, and so that, it can be really standardized. So he is really trying to put the clear, useful and fruitful dialogue between Ayurvedic practitioners and the understanding, so that it can be universally accepted. I really appreciate working with Ramesh Warrior, because we have been doing some work. I've been both as patient and as a collaborator. So I had the privilege of working with him and his co-workers, which is a very important team which is there in Madurai, and also in Coimbatore and in Chennai. So I think they have a very good structure. And he has got also a lot of new ideas, because Ramesh has come with a lot of new ideas, saying that maybe correlation between the modern scientific data and already the clinical data available in the Ayurvedic practice. So I want to listen to Ramesh along with all the participants. Will be really excited, because this is another side of the picture of how we can treat diseases with Ayurvedic system, which is a holistic approach. On to you, Ramesh. What happened?
I think my video is blocked.
Oh, yeah. What happened?
Yeah. Well, now it's coming. So, good morning, everyone. Thank you, Dr. Vijayalakshmi, for the wonderful introduction. Very elaborate, I would say.
Thank you. It's my privilege to be in this August gathering today. I see that most others are great stalwarts in the area of modern drug development and modern pharmacology and drug development. So I'm sort of, fools there where angels fear to tread. I'm from that perspective, but nevertheless, I would definitely like to throw in another perspective about how Ayurveda looks at the whole spectrum of immunity. And while the previous, the whole focus was on the people who are on the drug development side, on the regulatory and so on and so forth, I think my talk will add a lot of value to what a common man can today do and how they can seek help from the traditional wisdom that's available in India for centuries, which would definitely help at this time when nothing is proven so far. So I think a lot of my talk would help in addressing the needs of common men. At the same time, also have with it the potential foundation for development of great drugs, provided the scientific machinery of the modern science is put to use to validate, as my previous speaker, Dr. Ramchand, was speaking. Maybe from the reverse pharmacology point of view, if this could be leveraged, I think it could be a great opportunity for our country to be at the forefront of this battle against COVID-19. So with this brief introduction, I would get into this. So my talk is going to be more conceptual in the sense of trying to get this audience familiar with really what Ayurveda is talking about. I think very often Ayurveda is misunderstood to be a collection of herbal remedies. But I would like to present a slightly different variation from that. From a conceptual perspective itself, Ayurveda seems to have understood immunity from a slightly different perspective, which I think also makes a lot of sense, because the way I look at knowledge is two-pronged. One is a very microscopic view of everything where you know every tree. There is another perspective which is a macroscopic view of knowing the jungle as it is. I think the mutual strengths between modern medicine and traditional medicine have been that, especially in Ayurveda, we have a very macroscopic view where we reduce a lot of minute things into one common concept. And that provides certain strength at certain avenues in certain concepts. Whereas I do agree that there is a very important role that a very fine, a reductionistic approach where you know every tree in the jungle can give us a different perspective. I personally am of the belief that you need to integrate the macroscopic and the microscopic view to get an overall perspective of what we are dealing with. And that perspective, I think, would give a lot of clarity and a much better way of dealing with whatever problems we are facing. I think I'm going to address the whole session more from the macroscopic perspective, because at the microscopic level, I think almost all the other speakers are going to be dealing with that at the deepest level, at the specific protein, at the specific receptor, at the specific interaction, so on and so forth. I am looking at how Ayurveda looks at it from a much, much higher plane, higher in the sense more gross, more comprehensive. So that is the basic difference I would like to point out between, I'm not restricting my topic to just Ayurvedic medicine, but Ayurveda in totality, the way it looks at immunity and how it believes one can enhance immunity. With this introduction, I get into my talk. So immunity in Ayurveda. Immunity, like in modern medicine, is also a concept in Ayurveda. We define it by a term called Vyadhi Kshamatva. Vyadhi Kshamatva is the ability of the body to handle diseases. One is its ability to prevent them, and two is its ability to overcome them. So Ayurveda's definition of immunity is not limited to fending off external intrusions. As we know it today, when we talk about the word immunity, we primarily talk about our ability to fend off external invasions. But from the Ayurvedic perspective, when we talk about, we don't have a word immunity in Ayurveda, incidentally. We only have a concept called Vyadhi Kshamatva, which is our ability to prevent diseases. And that means it's not just fending ourselves from the attacks from an external intrusion, but also the ability of the body to prevent illnesses of any type. That is, there are intrinsic changes that are happening due to the metabolic process and several other factors which I'll be discussing about, body itself has intrinsically a tendency to create diseases, as we know of them today as non-communicable diseases. And the body's ability to overcome an illness if it were to manifest. So the whole spectrum of how do you prevent an illness, and second, if it were to manifest and it creates some sort of change in the body, how do you get back to normal? So this whole scope is considered under this concept of Vyadik Shamatva, which can be equated to immunity. While there was a clear understanding that external factors do cause illnesses, so there Ayurveda did understand the role of external. I'm sorry that there was a small interruption. I hope the line is clear again. So while there is a clear understanding that external factors do cause illnesses, the greater focus in Ayurveda has been on the internal state. How is the body capable of fending off this thing? So though there are external organisms, we all know that external invading organisms are in abundance. They are always present around us in whatever we eat, in what we breathe, and so on and so forth. But the real critical issue is how is the body able to respond to that? How is it in a state of equilibrium and homeostasis? And how is that able to prevent these invading organisms to create trouble within the body? So the determinants of health as described in Ayurveda are predominantly in terms of proper structural and functional integrity of not just the body alone, but the body, mind, and soul. There is increasing work, modern scientific work, that has gone to demonstrate the role of the mind in every aspect of the body's functioning, not to talk about just immunity. There have been several works that have been done where one has been asked to visualize a particular way, and they were able to demonstrate that immune markers changed dramatically with a type of visualization and meditative process. So there is enough evidence today. There is no more the need for us to speculate that mind has got a role in the functioning of the immunity. There is enough scientific evidence to tell us that the functional and structural integrity of the mind, body, and soul is what constitutes health, according to Ayurveda. And anything that upsets the balance beyond its tolerance limits was understood to cause disease. So it's not a static state of balance as we might generally tend to think. It's a very dynamic state which is continuously fluctuating with so many factors. So it has got the level of imbalance which can be tolerated well, up to which we don't call condition a disease. So there is some level of, you don't feel hungry. I mean, you can call it a disease, but it is an imbalance. You don't feel like sleeping. You feel tired. You feel so. There are so many small things that can happen which need not be labeled as a disease. They are self-correcting. So there are levels at which the imbalance is created, which has got a tendency to get back to normal. But at the same time, if the balance is upset beyond a certain critical threshold, then it is known to cause disease.
Sorry, teacher, sorry.
The body is described as having an ability to ward off diseases if its structure and functions are in a state of balance or homeostasis. So I think this is the fundamental concept that Ayurveda undertakes to talk about how one can maintain health. So immunity from that perspective is a much larger concept than just the adaptive or the specific immunity as we call it. So there are several factors that are known to influence this homeostasis, which is described as always being in a state of flux. So Ayurveda understands that the functional aspect of the body is continuously changing. There are several factors which affect this sort of a structure, this sort of balance, and they need to be factored into. Some of those factors are the genetic propensity itself. Ayurveda understands the concept of prakriti, which is about each person has got a genetic propensity to have a certain function in excess or slightly at a lower level. So that is one level of very, very fundamental thing that creates an imbalance in the homeostasis. The time of the day, the age of the person, the place where he's staying, the climatic conditions, the geographical conditions, so on. For example, a person living in the poles is exposed to a completely different set of things, and his body's metabolism is completely differently adapted compared to a person who is living in the tropical zone and so on and so forth. A very marshy place is different from a very dry place. Food, the type of food that he's eating has got a huge influence on the way his hemostasis is balanced out. Eating food immediately changes the way the body works, and few hours after your food intake, the body is different. Sleep. So if you do certain things just before food, that can be a different effect, and if you do it long after you take food, it can have a different effect. Sleep. The activity. If a person is very active, it has got a different balance. The balance of the functions in the body is very different from a person who is very inactive. I mean, I'm not just talking about black and white, and all shades of gray in between have got to be looked into. The state of mind has got a huge role to play in the homeostasis that the body maintains. His daily routine, as we today know of the biological clock, which needs to synchronize with the chronological time. So this particular circadian rhythms that we develop, and if you are in tune with the biological, with the chronological clock, it is one of the very important aspects of health, which itself is immunity according to Ayurveda, because a full state of proper state of health itself is the greatest immunity that one can have. I'm not talking about a very specific ability to ward off a particular infection, but immunity in general. The daily routines, the seasons, every season has got a different effect on the body. You can see that several diseases aggravate during certain seasons, and they get better naturally during certain seasons. The environment. Today we talk a lot about the effect of the environment on the health. This is an understood topic today, and we also know about the effect or the negative effects of toxins. So toxins is a very important topic in Ayurveda. We talk about toxins that are exogenous, that get sort of internalized into the body through the different sources, but also toxins that are self-generated by the body as part of its normal metabolism. So we produce a lot of toxins which can get accumulated in the body and which can cause diseases from within itself, or unknown external factors and so on. So we know of certain things, I mean, like, infections and so on. Those days, there was no clear understanding of different types of microorganisms. We didn't know about bacteria or viruses and so on and so forth, but they definitely knew that there are external factors that also cause diseases. So, I mean, this is not a comprehensive list. This is just a list that can give us an idea about how many factors need to be considered when one is talking about how do you prevent diseases from happening. So the one thing I would like to leave at the end of this discussion is that it is a very multifactorial understanding that constitutes the way towards maintaining proper health. We define all this through a concept called Urges. The word Urges is mentioned in Ayurveda, which epitomizes the ability of the body to preserve its health as well as ward itself against diseases. So this is described as the most important factor that helps prevent diseases and helps an individual to stay healthy. So Urges is definitely one of the key elements of immunity in Ayurveda. So if we were to fairly correlate, what are the components of immunity that we talk about in Ayurveda? This is one concept which encompasses all these different factors. So if you have to say that Urges is good in its perfect state, I would say that most of the components of the immune system and several others beyond the current description or understanding of immune system have got to be in place for one to say that his Urges is up to the mark. So body keeps replenishing the Urges as a part of its normal functioning, and its depletion due to several causes which I will describe later leads to diseases, both caused by external factors. We are easily prone to infections and external factors causing diseases when our Urges goes down. So also we are also prone to internally triggered diseases. Today we are knowing more and more about the role of our own immunity in the not so infectious diseases as well. Today we know of even the neurodegenerative diseases as an immune response to toxins that are getting internalized through the gut microflora and so on. So several new concepts are emerging, and we know that the immune system has got a role to play in almost any sort of disease manifestation. And the protective function I think is still, we understand a lot in bits and pieces, but as an aggregated whole, Ayurveda tries to put it across as a concept called Ojas. So when we were to balance this finite knowledge of each of the components that go into making a person healthy, to help him reverse his disease process and so on, we would be explaining Ojas in greater detail. So I would think all of modern immunity understanding is adding value to the understanding of Ojas from that perspective. So there is a concept of depletion of Urges. Ayurveda says that Urges gets depleted due to several factors. Number one factor that is mentioned in Ayurveda is psychological stress. When a person is in a negative frame of mind over a large period of time, we are very well capable of having acute episodes of psychological stress. But if it were to persist in a chronic manner over a long period of time, that is a very clear cause of depletion of Urges, which can be translated to not adequate immune function. Not appropriate, I would say, not even not adequate, not appropriate immune function. Poor dietary intake. If the nutrition is not proper, that is what constitutes proper nutrition is a big chapter again. But I would summarize and call here that proper dietary intake, if it's not there, that is understood to be a cause of Urges. Today we know that nutritionally deficient people have got a poor immune system. Excessive activity. If one is perpetually overworked and is not balancing his activity with rest and good sleep, that is a cause of depletion of Urges. He becomes prone to immune related issues. He is prone to diseases if he is not able to balance himself between the level of activity that he is involved in and the amount of rest that he gives to his body. Urges can also be depleted by injury and tissue damage. So certain diseases as well as external causes of injury, like external causes of disease like an injury, can also lead to reduction of Urges. So your immune system suffers when there is tissue damage. External factors like infection come also as a factor, but slightly lower down in the priority order according to Ayurveda. Chronic diseases are also known to have their toll on Urges. So a person with suffering from a chronic disease is affected by inadequate or inappropriate quality and quantity of Urges, which leads to his further propensity to get diseases. Advancing age depletes Urges, and that is why Ayurveda has a whole speciality focused on slowing down the aging process. One says you cannot completely prevent the aging in toto, but there is a very scientific way of how you can age appropriately and prevent premature aging. So aging is known to deplete Urges, and last but not the least, a very important cause of depletion of Urges is the toxins, both from external origin and those that are produced intrinsically by the body. Ensuring good Urges to ensure enhanced immunity. So what are the concepts that can be used to enhance Urges? They are more or less the complementary of what we said in the previous slide. So proper nutrition and dietary habits form one of the very important concepts in Ayurveda to maintain proper Urges. So that's why there's a lot of importance. Today we see that, holistic advice tells you about how you have to eat properly. Eating properly is a big chapter in Ayurveda, which is again not one size fits all. Food, the biggest controversy around food today is about this is good and that is not good. I think most of that is wrong, because no food is good for everyone and no food is bad for everyone. Each person's nutritional requirement is slightly different, and there are several factors that needs to be looked into before one can decide what is appropriate diet and nutrition for a particular person. So I think if we put that context into perspective, most of the controversies surrounding diet and nutrition today would be cleared off. There is a lot of imbalance happening. One day, one particular food is considered great food. After some time, the same thing is touted as the worst sort of food for human beings. This is all because there is no context in which this particular statement is made. So Ayurveda gives a beautiful context to this whole understanding of nutrition. Then comes a healthy lifestyle. As I said, one has to balance between activity and rest. You cannot have too much or too little of either. You need to also have a sort of a routine, because body likes to have a routine. If you're constantly changing your habits, that is a bigger stress on the body, and that itself tends to deplete your ability to fend off diseases. Very important is that balance between rest and activity, as I said. A very pleasant state of mind, which again talks about the psychological factors. One has to be in a state of positive frame of mind. That has got a huge impact on every single cell in the body and the way it responds. Because stress, as we know, is just one of the triggers which are still existing in us from our cave times or the jungle times. The flight and flight response, which is the cause of most of the diseases of today's world. And this is not only applicable for the NCDs, it is very much pertinent to the infective diseases also. You can see that children who are extremely stressed due to their examinations, most of them get sick and fever and infections prior to their exams. Appropriate herbs and therapies to balance the bodily functions which get deranged. So as I said, the body is functioning is in a state of flux. It is constantly in a dynamic state, and it needs to work within a particular range beyond which it causes diseases. So when you find that the balance is getting tilted towards any particular direction, then you need to sort of balance it back. This is what Ayurveda talks about as treatment, the primary treatment, primary prevention, as I would call it. For example, it could be a small manifestation as if you don't have hunger. So that's a signal that the body is giving you that my digestion is not in proper state. So you need to take care of it. Maybe skip a meal. Maybe don't take anything heavy. Sometimes if it is too bad, you're feeling like throwing up, please do. Don't block that. Don't pop in an anti-emetic. If you were to purge once, don't immediately take one, anti-purgative. You're not supposed to, intestinal motility arresting drug is not to be used the moment you have one purge. For example, so there are several concepts that go into primary prevention. Small derangements that are happening in the body is signaling to us, and we need to take care of that. And for the toxins that inadvertently get into the body, Ayurveda describes a very elaborate mechanism of detoxification. Each of these are seminar topics by themselves, so I'm just touching upon the basics here. And last but not the least, Ayurveda talks about a whole specialty called Rasayanas. Rasayanas are herbs and herbal preparations that help to rebuild the urges. So the ability of the body to fend off diseases as well as to repair the damage that is caused to disease is enhanced by the use of the herbs and herbal preparations that we call as Rasayanas. So what does Ayurveda talk about as diet for immunity? Because as we said, the first factor that we spoke about is diet. Ayurveda recommends several factors that have to be considered in order to call a food as an ideal one. So it's not whether there is it's not a question of just whether you have enough protein, carbohydrates, fats, minerals, vitamins alone. Ayurveda talks about each individual's constitution. Each one of us are unique in our own way, and our nutritional requirements are different. Each state of health, if you're sick, you need a slightly different type of food and medicine as opposed to if you are sick. If you're aged, your nutritional requirements are different from when you're young, when you're a child. The level of activity also determines the type of nutrition that you need. Your state of mind has got a role to play in the type of nutrition that you need. What is eaten is just one of the components that determine the appropriateness of the diet. The quantity, the same food may be touted as good, but if you take a large quantity, the effect may be just the opposite. So the quantity, so all these factors have got to be interwoven to talk about something is appropriate or inappropriate. The way it is processed, it could be a very good dietary item, but if you process in a certain way, it could become deleterious to health. The combination of ingredients that go into it, something may be very good by itself, but if you combine two of them, they are not very good. They create a negative effect. The place of origin, Ayurveda very specifically says that there is a genetic adaptation to a particular type of diet and lifestyle, like dietary regimen. We are not very well adapted to suddenly switch over to a diet pattern that is completely alien to us for generations. The body takes time to adjust itself to a particular type of nutrition. I think we can all understand that a person from the south can never take as much sweets as a person from Rajasthan or Gujarat can take. Just one bout of taking so many sweets and you will throw up. and vice versa. Nobody can take as much chilies as an Andhraite can take because they are genetically used to taking that sort of food. So the place of origin and your place of origin, they have to be in match. The time of the day, certain foods are good to be taken at a certain time of the day, but not all the different times of the day. The season, a food is good in a particular season, but not so good in a different season. I could go on to give examples for each one of this, but I think it will be too much. The time will be a constraint. So I'm just rushing through the just main points so that we get an idea of the complexity involved and how it needs to be so personalized in order for us to maintain our health in an ideal situation. The season and what one is used for generations. So we call it a satmya. What we are used to eating for generations is naturally acceptable by the body. If you eat something, try to eat like an Eskimo, the next day you will get sick. You can't digest that sort of food. Food that meets these criteria helps improve health, strengthens the tissues of the body, and improves resistance to disease. So when we talk about immunity in Ayurveda and the ability of the body to withstand the diseases, you need to have nutrition that takes into consideration all these different factors and is ideally suited to you as a person. So that is where today's healthcare paradigm, which is about personalized medicine, is what is totally enshrined in what Ayurveda talks about as Ayurvedic medicine. Lifestyle, daily routine and regimen. Ayurveda says that you need to have a routine every day. You cannot wake up at a different time every day, eat at a different time, go to sleep at a different time. This is very, very harmful to the body. Body takes a lot of time to readjust itself. And the biological clock alignment with chronological time is also very critical. One sleeping throughout the day and then working through the night is not doing the best thing for his health. Today we know that the telomerase and all that gets affected by your switching time zones and so on and so forth. So all these different concepts that have been mentioned in Ayurveda are getting validated through modern scientific research today, as more and more people are putting their focus on these concepts. Balance between activity and rest, I already spoke about. This also has to be fine-tuned to suit each individual. So it's not a person who is a Vata Prakriti cannot sleep 8 hours a day. He will naturally be able to sleep only for 5-6 hours. But a Kapha Prakriti person needs about 7-8 hours of sleep, and so on and so forth. So it's not also one size fits all. A sattvic state of mind, being in a calm and composed state of mind, is a very critical component to help improve the immune status of the person. And a lifestyle that is adapted to suit the seasonal influences also is very critical for, especially in climates which have got the extremes. One needs to adapt his lifestyle to suit that particular climatic condition. Now we will get into the direct topic of Rasayana, which is the herbs. So herbs are just one of the components that go into the whole advice that Ayurveda gives in how a person can remain healthy and prevent himself from diseases. So several herbs and herbal preparations have been described in Ayurveda as Rasayana. Those that help develop and maintain the tissues and urges. So that is what it means. Ayurveda talks about the Rasayana as something that helps to build appropriate tissue. That means structurally competent and functionally competent tissues. That itself, the sum total of all that is what Ayurveda talks about as the concept of urges, which itself constitutes the ability of the body to be able to fend off diseases. So these Rasayanas can help specific tissues or systems. Many of them are like that. There are many Rasayanas that are specific for certain types of tissues. For example, respiratory, neurological, cognition, skin and skin appendages, digestive, reproductive, metabolic. So there are Rasayanas which are skewed towards preventing diseases as well as improving the resistance to diseases of these systems. There are also Rasayanas that are suitable for different constitutions or different pathologies. Rasayanas for Vata, Pitta and Kapha predominant people and condition. So Ayurveda understands the functioning of the body in terms of 3 factors called Vata, Pitta and Kapha. And their mutual balance is what is considered a state of health. So diseases are caused by either of them or 2 of them getting into excess, out of balance. And that imbalance has to be corrected. So we understand diseases in terms of whether Vata is predominantly a problem or Pitta is or Kapha is, or Vata and Pitta and Kapha, Pitta and Kapha and so on. So Rasayanas that are specific for these types of diseases as well as for people who naturally have a genetic propensity for a higher level of these doshas. So there are Rasayanas that are suited to those people, suited to those particular conditions. Rasayanas that are anabolic. So for example, earlier days, lack of nutrition was one of the biggest problems. I think even today, we are only having too much of fat, but not all the other tissues, not the other, tissues of the body. So there are Rasayanas that are essentially anabolic in nature, which help to rebuild those tissues that have been inappropriately developed. Rasayanas that help correct chronic diseases, because any chronic disease leaves an impact on the tissue and on the immune system. So there are Rasayanas that help you to rebuild the strength and your ability to ward off further diseases. And those Rasayanas are also there. And on top of that, there are general Rasayanas, those herbs that are suitable for almost everyone and can be used in all conditions. They are like your general maintenance stuff. So, there are these different types of Rasayanas that Ayurveda mentions. So some of the general Rasayanas is what I would touch upon. One is Amalagi. Phyllanthus emblica is one of the foremost Rasayanas and is known to retard aging. So one of the very, very important things that we talk about is how do you prevent aging? Because aging itself is known to be one of the biggest diseases. So Amalagi is one of those Rasayanas that can be very anti-aging. And then comes Triphala, the three fruits. One of the only things that is recommended for everyone anytime. It is helpful in healing all types of diseases. It helps in improving cognitive functions, longevity and memory. Specifically good for vision and to heal non-healing wounds and ulcers. We know today that they are so rich in antioxidants. A whole array of antioxidants are present in Triphala, in addition to several other chemicals. Devanaprasam is one of those herbal preparations which is based on Amalagi or Phyllanthus emblica, and the sort of indications that it can be used for is mind-boggling. It is specifically touted as being very helpful to improve immunity. It helps in the relief of respiratory problems, inflammatory arthritis, fevers, heart diseases, urogenital disorders. It is an anabolic. It improves cognitive function, memory, skin tone, prevents diseases, improves longevity and functioning of the sense organs, and is also an aphrodisiac. So it's like a panacea. I mean, it's not that it can cure all the conditions like that, but it is helpful in all the different conditions. That's what it means. Brahma Rasayanam is another very common drug, common Rasayana which can be used across the board, which contains Haritaki, which is Terminalia cebula, and Amalagi, which is Phyllanthus emblica, as the main ingredients. It is also known to help reverse all signs of aging, and it helps to improve longevity. Incidentally, Amala Cancer Center, Trichy, has done a research on its effect and role in cancer. So there are studies being done on all these different products, but not to the extent of, the way a modern drug is studied, because the sort of resources that are applied to this research is far less compared to the sort of resources that pharmaceutical companies are able to plow into the research of modern drug and the market potential. This is far more complicated to handle as a commercial product as well. Rasayanas with specific system-specific action. For example, there are lots of Rasayanas that are very specifically helpful for the CNS. Mandugabarni or Hydrocortyl asiatica, St. Madhu, which is Glycyrrhiza glabra, Guduchi, which is Stenospora cordifolia, Shankapushpi, which is Convolvulus pluricalis, Brahmi, which is Dacopa muniera, Vacha, Acorus calamus. These are all single herbs that are very effective in improving the immune system as well as in reversing diseases pertaining to the CNS. In reproductive health, neurology, and strength, there are several herbs that are mentioned: Nagabala, Sida veronicifoliae, Gotrura tribulus terrestris, Ashwagandha vitania somnifera, Dietary, which is Proraria tuberosa, Shatavari, Asparagus racemosus, Bella, which is Sida cordifolia, Atibella, Abutilon indicum, and so on. So these are all very good for the neurological system in reproductive health as well as strength-giving herbs and so on. So like this, for every system, we have got herbs, Rasayanas that are specific for the specific system. Other herbal Rasayanas, there are Chitraka, which is Plumbagos elanica, is very effective in all the Vata-related conditions. So Vata implies skeletal-muscular and neurological diseases predominantly. So Chitraka is one of those herbs that is useful in all the Vata-related conditions. Ballataka or Semicarpus anacardium is a Rasayanam that can be used for Kapha-related diseases. Tuberaka, which is Hypnocarpus laurifloriae, is another Rasayana that is mentioned for all types of skin conditions. Pipali or Piperlongum is a very good Rasayana for respiratory diseases, diseases of the liver and spleen, and in several autoimmune diseases. Shunti or Zingiber officinalis, our common ginger, is very good for respiratory, liver and spleen, and autoimmune diseases. Haridra, Curcuma longa, which is one of the most highly studied Indian plants. I mean, as Dr. Ramchand was speaking, so many patents and so many discoveries have come out of this one wonderful Rasayana. Haridra is touted to be very good for respiratory diseases, liver, autoimmune diseases, diabetes. Somaraji or Sorelia corallifolia is one of the wonderful Rasayanams for all skin-related disease. Lashuna or Allium sativum for all types of Vata-related diseases, but for persons and conditions not involving Pitta. So while everybody says garlic is a wonderful herb, it may not be suitable for long-term use for everybody. Pitta people with the Pitta condition will aggravate their Pitta by using garlic in a large quantity over a long period of time. Silajato, which is asphaltum punjabianum, it's actually still there's a controversy whether it is a mineral origin or if it's a plant origin. It is primarily found by the degeneration of organic substances on mountains. It's like a tar-like substance. This is very, very effective for renal and a host of other problems. So that is also Rasayana. So these Rasayanas that I have mentioned above are all very powerful Rasayanas. And the way we use it as a powerful Rasayana is just a large dose of just this single herb. Large dose, it has probably as a meal with nothing else. Each one of these Rasayanas has a specific dietary regimen to be followed. And this is all done only after a proper detoxification is done. So this is the way we use a Rasayana as a very potent medicine. So very rarely does Ayurveda recommend a single herb for any particular condition. And if it is used as a single herb, generally it should be used as per the Rasayana methodology. So where it has got a very profound effect on the body when given at that level of dose, but at the same time before that one has to subject himself to a detoxification program. Otherwise the effect of Rasayana is dulled out. So this is a process called Pudi Praveshikam, which is a more intense form of Rasayana, which is done with all these different herbs that I mentioned above. Now I will come to the Rasayanas that can be used without much restriction. Amalaki is one of those Rasayanas, that is Emblica officinalis or gooseberry is one of those Rasayanas that can be used without any restriction. As I said, anybody can use Amalaki. Haritaki or Terminalia cebula. Even Ayurveda describes how to use each one of these and in what conditions, what sort of benefits it would give us. Punarnava or Boyaravia diffusa, very good for renal conditions. Brihati or Solanum indicum is also good for renal conditions as well as a neurological tonic. Amshumati or Desmodium gangeticum is very good for Vata, especially conductive deficiencies of the heart. Asana or Thyrocarpus marsupium is very good for metabolic diseases as well as for the skin. Shatavari is very good for Asparagus racemosus. It is also galactogog. It is also got functions of reproductive system. It's also good as for the cognitive functions. Ashwagandha is one of those highly touted Indian herbs, which is primarily an adaptogen. It is also an anabolic. It has got an effect on several diseases. Vitreloids today have been studied, and they are also being touted to be one of the things that could be very effective in the fight against COVID. It is one of those herbs that have been positioned as COVID. There are a few research papers but most of them today still at the level of in vitro studies or animal-based work. Today, just now we have got the opportunity. I think some clinical trials are also happening on the effect of some of these in COVID. Sodamstra or Mucuna pruriens is a neurological tonic and a Rasayana. So these are some of the Rasayana single herbs that have been recommended with a specific protocol on how they can be used as Rasayanas. Traditional herbal preparations.
Dr. Ramesh, I think we have to wind up now. Yeah.
All right. So I will just go into a couple of slides, 2 minutes. I will wind up my talk.
Yeah.
So modern research findings on some of those herbs. I will now go into just the level of infections. So when we talk about the infectious diseases, these are the herbs that are mentioned: turmeric, amla, st madhu or Glycyrrhiza glabra, Piperlongum, Tinospora quadrifolia, Allium sativum, Andrographis paniculata. And these are some of the herbs in addition to ashwagandha which are being currently evaluated for infectious diseases in specific to the viral COVID-19 that we are talking about. There are quite a few studies and the chemical components that are part of these herbs, which are now being seen to be effective, and their mechanisms of actions as elicited from in vitro as well as in vivo in animal studies is what I'm giving here. So I've shared some of these that could be seen from my slides. So Curcuma longa is now touted to have very strong antiviral effect. Allium sativum, the essential oils of garlic have been known to have a binding on the SARS-CoV virus binding sites. Virucidal effects of Allium sativum have also been already expressed. Embolica officinalis, the major phytoconstituents are there, and it is very widely known to have effect on so many of these, cytokines, TNF alpha, interleukin 6, interleukin 1b, COX2, IL, and so on. So COX2 inhibitors. So several immune-related functions of several of these herbs have now been studied and reported. Glycyrrhiza glabra is also being touted as one of the very good potential herbs that could be effective in SARS today. Glycyrrhizin is known to block the replication of SARS RNA. Major phytochemicals in piperlongum, it is primarily something that enhances the bioavailability. India has already pioneered the work of how to use piperlongum as an adjuvant to anti-tubercular therapy, and how we are able to reduce the duration of the therapy by adding piperlongum. So it's very effective as a bioavailability enhancer. Tinospora cordifolia is a very well-known immunomodulator, and several of those compounds are known to have strong immunomodulatory effects. Titania somnifera, as I said, it is predicted to interact with the transmembrane protein serine, and it is touted to help block the entry of SARS-CoV virus into cells. And several of these studies are today reporting about the effect of some of these Rasayanas in how they might prove to be potential help in working with patients with COVID. Andrographis paniculata has already been studied very well, especially in the context of dengue. And they also found that several of these natural compounds can be structurally modified to make them more effective and more specific. So there is a work that has been done on that, which is exhibiting how some of these are able to have a specific binding affinity towards the spike glycoprotein of SARS-CoV virus and ACE2 inhibitors, and may be useful as therapeutic or prophylactic agents. Thank you. I hope I have covered the conceptual part more than this, because otherwise most of that is in the public domain. We all know about the herbs that are being studied. I thought it was more critical to share the conceptual understanding of Ayurveda. Thank you very much for the patient hearing.
Yeah, thank you, Dr. Ramesh. I think you really gave a good, I would say even a big course to understand the concept, and then from there how to take it to the modern application. Only thing is, many of them may not know the what you call vernacular name of the plant which you use in the Sanskrit version. And I think maybe you should give us a reference of some dictionary which will give the vernacular versions of all these names, like for example, Amlika is Nelikai, and like that. So that will be very useful.
Yeah, I will share the vernacular as well.
Yeah. I think that's a very interesting talk. I think also, as you mentioned at the end of the talk, that you are going into the components which are doing, and that is probably one of the work that we are also doing together with you in terms of understanding what is the pathway, which component is doing what. So I think this is a very interesting exposure to something that people know from a distance. They do not know what it is. And I think more and more people should come as an interface between biology and Ayurveda, so that we just make Ayurvedic thing more useful and more open to the world, larger number of people. Thank you very much. Maybe the participants have questions.
That was a very good talk, Dr. Ramesh. Thank you. One question from the participant is Urges an Ayurvedic analog to the allopathic reference to antibodies that the body produces?
No, not exactly. It doesn't restrict itself to just the antibodies. Urges is a conceptual understanding which encompasses everything that can contribute towards preventing diseases. So Ayurveda mostly talks about concepts. It doesn't try to identify just one compound. If you try to say antibodies urges, you are limiting the scope of urges. So it is antibody and all your other things, including the antioxidants and the whole cascade of substances that go in helping the body remain healthy.
Okay.
Toxin.
Dr. Sabrish,
Yeah.
Due to time limit, we will not take much question.
Okay.
So we will take 10 minutes of a break and then we'll come at 12:10.
Right. Okay. Thank you, sir, Dr. Ramesh. Thank you, Ramesh.
The break, maybe people can talk. Hello, Sanjeet. During
yes,
the
yes.
break, if the participants want to discuss with Dr. Ramesh, we can take it because break can
yes,
be,
yes.
yeah.
Okay, ma'am.
Because
Okay,
maybe lot of questions will be there, because lot of disputes with Ayurvedic, so maybe lot of participants have questions.
Yes, ma'am. We can take the question.
Yeah.
Okay, Sabrish.
Sabrish,
Yeah.
go ahead.
Okay. Okay. Then, okay. Two people have asked the same question. How psychological stress deplete urges?
Yeah, today we know that stress is directly working. So what happens in stress, as we know, is that we are sensing a stage of an emergency. When we are stressed, the body assumes we are in a state of emergency. So the entire priority for the body is to either fight or flight from that particular situation. So the entire dynamics of the body is skewed towards those processes that enhance your ability to fight, which is you need more oxygen, you need more glucose. So you need your circulatory system to be working at its peak. Your heart rate goes up, your respiration rate goes up. You produce more glucose in the body. You produce steroids in the body. As we know, so the whole dynamics of the body is shifted towards just energy production. At the cost of several other things, body has limited bioenergetic efficiency. So if you are...
Hello. What happened?
Demanding too much, what is happening is those functions like your reproductive system, your immune system, even your digestive system, and all those other things that are not very critical in a crisis like an emergency are shut off. So this is what is one of the most important things that happens in stress. Your immune system is severely compromised because the whole biodynamics is shifted towards producing energy and providing oxygen. So if it is happening in a short period of time, there is nothing, not much of a problem. Body already rebalances itself. But chronic stress is the thing that really creates a complete upset. It is known that places that are torn by war or serious psychological stress, all these functions, the reproductive rate also comes down. People are prone to get sick naturally because of stress, because it suppresses. We know steroids are immunosuppressants. When you are constantly producing too much steroids in your body as a result of the stress response, which is known as the hypothalamus pituitary adrenal axis. So with that, you secrete more of steroids. The steroid itself acts as an immunosuppressant. So you are not able to respond to any infection the way you are otherwise capable of. So that's how psychosomatic effects are produced, more importantly, with respect to immunity.
Okay, one last question, sir. What about the heavy metals present in plants? What about the heavy metals toxicity in plants?
One of the biggest clarifications I would like to bring about is a lot of this controversy is not related to heavy metal presence in plants. The bigger problem is heavy metals being used as medicine itself, along with plant material. So there was a whole branch of Ayurvedic medicine which came in much later, where the use of heavy metals also came into practice. But they were known to be toxic. The very name for those materials was Pashana. Pashana means poison. So they knew they were poisons, but they had a method of preparing those in such a manner. Ideally, if you prepare a metallic preparation, it should become a nanoparticle. The bhasma, which is typically prepared, is supposed to become a nanoparticle, where it doesn't really then obey the rules of a normal metal. But the quality control methods, those have been mentioned, those days were more subjective, and people did not often follow that. So what is happening is inadvertently, it is not becoming the type of bhasma which was supposed to become. and then it is being used as if it is a very normal herbal medicine. So there is very little understanding among the public about what is herbal medicine and what is herbal mineral preparations. Lots of Ayurvedic medicines today are directly on the label itself. The mineral composition is also given there. But people don't understand that. So very often when that is being used like a herbal preparation, there is a lot of metal toxicity that can come in. The next part is the inadvertent metal that gets incorporated into the product through the processing. Also the plants, some of those plants have got a tendency to absorb heavy metals from the atmosphere as well as from this earth. So today with modern techniques, like, we have got the testing methods, you can do a spectroscopy and so on. If you can test your raw materials and finished products, we are able to ensure, with proper quality control, we are able to ensure that products are free from heavy metals. We've been constantly testing these. Our products are being exported to Germany and so on and so forth. And again, it is subjected to test and they're all coming clean. So it's not that Ayurvedic medicine is always adulterated with, or it's got the heavy metal contents. It could be inadvertently there because even small time players are allowed to operate in this industry, because though GMP has become mandatory, it's not always, as we know in our country, the implementation is not always as it is desired to be. So there is inadvertent thing on one side, but more importantly, most of these reports are around products that are purely herbo-mineral products. And but only thing is that is being exported abroad as a dietary supplement. And there they come to find that, they have got very high levels of metals, and that is causing all the controversy and bad press to the Ayurvedic system of medicine. So I think the government of India is taking steps to regulate that. We are now supposed to test our products with a third-party lab, which is GLP accredited, before we can export the product. So government is trying to do all of that, but we are experts at also finding some bypass, which is what is sometimes creating a bad name for our country. I hope that particular thing, it can very well be overcome. With conscientious effort, today we have the technology to ensure that Ayurvedic products can be free from heavy metals, and we as a company are able to ensure that, and we are very happy about that particular claim.
Okay, thank you, sir. So with this, let us conclude this session, sir, and we'll go on to the next session.
Thanks again to Dr. Ramesh. Sir. I think probably what is needed, one comment. Yeah. What is needed is this should reach the people correctly with their clear understanding. That is the problem. The problem is the allopathic medicines are defined with single target, single medicine, whereas this is a holistic approach. So I think that is where we need to have kind of a dialogue between the two population and reach the common man in a better way, in a much better manner. So this is my wish, and with this I thank again Dr. Ramesh.
Thank you very much. Thanks for this opportunity. Dr. Krishnan, Dr. Vijayalakshmi, Dr. Shabarish, all of you. Dr. Narasimhan is also there. So thank you so much for all of you for the very good hearing. And I look forward to advice, comments, suggestions to improve on what we know further. Thank you so much.
Thank you, sir.
Sabarish, we will directly go for next session, okay? Yeah, no break. Okay. We'll go for directly for next
session. The break
session.
is over.
Yeah, break is over.
Okay.
We'll go. Yeah, yes, I will ask BG ma'am to introduce.
Dr. Narasimhan. Yeah, you just give the profile sharing. So I will introduce him. So is it ready? Yeah.
Yes, ma'am. It is ready.
Okay. So now I have the great pleasure of introducing one of my friends that I came to know immediately after I set up my center in Vellore in 2005. Dr. Narasimhan is one of the first persons who I had to help me. So I can be very frank. Dr. Narasimhan, first I will say who is he. He has done his PhD in chemistry in Madras University. I think he's Goindachari student. And after that, he was working, he did his postdoc with the Nobel laureate Brown in Purdue University. And after that, he was working in Speak Foundation for some time, which was also my dream to work. But by the time I was trying to set up my center, Speak Foundation was wound up. I think that is probably how he had a very close interaction with Dr. M.D. Nair, who used to appreciate him very much, because I know that for myself. And he is interested in, because not just doing research with Nobel laureate, but he is interested in taking to the better understanding of our herbs and our practices and how to bring them with the modern understanding. So he is trying to really understand the herbal products, and with that he has set up a very good structure called Astagiri Foundation, which is mainly involved in the herbal extracts and herbal extract medicinal value. And he is also involved in training. That he was the co-founder of the Chromatography Society of India. As you know, chromatography is a subject that has been neglected completely in the academic institutions. Also in other countries, but more in India. That was one of the reasons why I was brought back from France to India. The government told that this is a field we need. But still, I think we are still lagging behind in that. So Narasimhan was one of those who started that structure, but mainly for the small molecules, because their efforts are focused towards the small molecules. And that Chromatography Society of India is organizing a lot of training programs with HPLC systems and other systems and proteomic for small molecules. And he has been involved in different training programs and different organizations. And personally with me, he was the one first who suggested some of the difficulties that may happen in the aloe vera product identification, I mean the raw material identification. So with that personal note, I would request Dr. Narasimhan to give his talk. My no? You ask him to unmute. So can you unmute and then start, Dr. Narasimhan? No, we are not hearing.
Sir, you have to remove the mute symbol, sir. It is on the left-hand side, bottom left-hand side below, red color icon.
Yeah.
Yeah, yes, sir.
Okay. So
please share your screen, sir. Bottom side, green color icon.
Yeah, I started that, and okay.
Yes, sir. It is coming. It is in the process.
Yes, sir. Fine. Make it big. That's a full screen.
Yes, sir.
Yes, fine, sir. You can start, sir.
Okay. Thank you, Dr. Vijay, for a very nice introduction about me and our interactions earlier. And when yesterday you were telling me that you came to Vellore and got stuck, or rather you liked to stay in Vellore, I thought you were the other representation of IISc for Vellore. The recent IISc for Vellore now is you. So with this small comment, I would like to go through this talk, herbal intervention in the management of COVID-19. So what is this herbal intervention? We have been talking about the various aspects, or rather, this COVID-19 is unabated even now, and it is growing and growing. The one thing which is not controlled, and there are many attempts. There is kind of tame the same by any attempt, any means. So this is what is going on, and it's a good seminar that you have started on drug discovery, development, and also on drug repurposing, because this is going to be the quicker way of controlling the virus, or quicker way of getting a relief for controlling the virus. in this process, we have missed about the phytochemicals. So this is a direct drug, which is synthetic ones, and now we are going into the herbals. The herbals are mainly useful because they are directly, if it is found useful, they can be directly applied for the control. So now you have these phytochemicals. There are so many of them, antioxidants. There are hormones in the phytochemicals, preservatives, inhibitors of enzymes, and also DNA protectors, and so many kinds of molecules are present in the phytochemicals. And therefore, there is a good scope for going through this herbal area, herbal chemistry mainly, to understand what is happening with the modern concepts, and also take the cue from the earlier Ayurveda literature. Now, the drugs in use right now are, although they are not really 100% effective, you can see the kind of chemistry here. So one is a phosphorus compound. The other one is like a caffeine type of a molecule here, but not a caffeine. And you can see here a chloroquine, which is a quinoline type. And all these kinds of molecular structures are there. Generally, they have some kind of a pharmacophores, which are useful in binding with the protein targets of the virus or the host, so that you get some benefit. So all these things have got a certain kind of a function in the body. Now, COVID management drugs. So I'm not going to go into the details of this one. Rather, I'm not an expert in this area, but still, I would like to go through some of these chemicals which they have used, and then what is their function here in drug binding pockets, and what is that they do with the protein or the targets here, the ORF1 and NP. So many NPs are there, and how do these things dock with them and then do their function, either in aiding the host immune or in controlling the virus replication. So now this Ayurveda perception of this infectious diseases, Dr. Ramesh has given a very lucid explanation of all these things. And it is quite amazing to see in the literature, this Ashtanga Hridaya Sutra Stham. I found that they didn't have any kind of tools in those days to understand why certain things are happening, why people are dying in large quantities, large numbers. So they called this as a bhuta, evil spirits or bacteria, parasites, insects, and such microbes come under this term bhuta, which they don't know how to qualify. Therefore, the kind of diseases or the infectious diseases that happened in ancient days, they used to call that is due to bhuta. How do you treat them? So they have given some kind of prescription, treatment of diseases involving these pathogenic organisms. So this Charaka Samhita and strategy for management of this one in 3 ways. One is extraction or removal of the pathogenic organisms present in the body inside. And the other one is Prakti Vigata, creating unfavorable environment for the pathogenic organisms. That is, it goes to the kind of origin of the organism, and there you control that one. It is the environment, you make the environment unfavorable for the organisms to grow. Now we are using all the sanitizers and things like that. So that comes under this category of treatment for this kind of infections. And the other one is avoiding the causative factors. So how do you do that? One, by creating some kind of a immune models or immunity in the body. So this pathogenic organisms, methods to kill the pathogenic organisms, and they are calling them as crimigna or some pronunciation, I'm missing that. So this kind of treatment is recommended in Ayurveda, and they have also given a large number of prescriptions, or rather the number of herbs that can do this job, like Madana, Maduka, Lamba, Nimba, Bimbi, and all these kinds of products have been suggested in Ayurveda for the control of the microbial organisms. See, of this, one of them is this Vidanga, Medulla or Vidanga. And this Vidanga is supposed to be a Jeet, or a conqueror of all the microbes. So that means this is the Emboliya rice, which is a very important herb in this virus control. Now the other suggested plants are Gugulu, Vacha, Acorus calamus, Vidanga, Emboliya rice, and neem oil. And there are so many herbs are mentioned in Ayurveda for the control of virus, but not just virus, it is the microbials. So these plants are all, these herbs are the antimicrobial products. Now the molecules here, you can see they are, each herb has got certain kind of a molecule structure. Here this is a saponin type of a structure associated with a glycoside, which is saponin. And this Rauwolfia serpentina has got a lot of alkaloid structures, which are also somewhat resembling a carbazole alkaloid. And here this is another Ativisha, aconitum. This molecule has got very interesting organic structures, aconitin and things like that. And Caesalpinia sapon is like a polyphenol product, and this is also called a Kachaka. So these are some of the molecules that are present in the herbs. And so what is that happening in the market right now with the herbals? There are certain products already have been in vogue. People are using it, like the Kapasura Kudinir, which is made of about 19 herbs, I was told. And Nilavembu Kudinir is already there, for which has been used for dengue and all. And arsenic album is a homeopathic product, and Coronil. And this is, I think, my own company which has brought this out. And here, if you take this Kapasura Kudinir, there it requires a number of herbs, around 16 to 18 herbs. And Ramesh was telling that Ayurveda doesn't go for a single herb product. Normally, it goes with the multiple or polyherbal formulations. Yes, so in that concept, this Kapasura Kudinir has got about 16 to 19 herbs. The only problem will come later will be how you will be able to collect all these 19 herbs to give a product of the same efficacy. So if one or two herbs are missing, will it have the same efficacy or not? So this lends to some kind of doubts, and also kind of clarity needed in terms of evaluating all these herbs individually and then in combination. And Nilavembu Kudinir has been already been showing to be a good antiviral or antimicrobial properties. And arsenic album is like a placebo type of thing, in my opinion, because it doesn't contain arsenic. I got one sample from the market and I analyzed for arsenic. It is very, very, very low. It is less than the recommended or the upper level. So it is kind of a product which is made from arsenic, probably dilution, and final dilution. There is not any arsenic in this one, and that product is given, probably it gives you a placebo effect or a stress-free effect, which is helpful in controlling your other emotional things, and thereby it helps in reducing your other panic actions. And I'm not going to comment on this. So the efficacy of the compounds are evaluated through bioinformatics. Actually, there is no clinical studies. Now there are a lot of patients out there and they are doing it, but I have not got any kind of a record stating that this has done so much of improvement in certain parameters. medical parameters and all, or clinical parameters. So therefore, right now what they do is take all the 19 herbs and then take their molecules. Some of them are already reported. And so then go through the bioinformatics and say that this is the mode of action. So in that way, I found that in Commercialization, one of them is that Tinospora cordifolia, that is the important herb which can be responsible for this efficacy. These studies indicated that pharmacophores of the active molecules as containing ketones, polyphenols, tannins, flavonoids, and steroids. From all these kind of herbs, you can see that the active molecules are represented as ketones, polyphenols, tannins, and flavonoids. So now what do we do? What is Astagiri's attempt in this scenario? So we had a product developed as a TSAM for management of acidity and support drugs like metformin, and it follows the concept of anupana described in Ayurveda. Anupana, you must be knowing that it is a kind of a vehicle for carrying the drugs. So this was a product which we made some time back, and then this was being consumed by people, and there were some good reports. And we also found that when this product was left for storage stability, we found that the aqueous extract was not contaminated by any microbes even after 2-3 years. So that gave us curiosity to check that what is present in this extract. So therefore we analyzed for the polyphenols and flavonoids present in this extract using HPLC method. And you can see here, this is the kind of chromatogram we get for the extract. And each peak you can probably analyze or identify based on the UV spectrum that you get. So one of the retention time here that gives a UV absorption spectra like this, which is definitely identical with mangiferin. So this extract has got mangiferin in it, and epigenin. Is this like a flavonoid UV spectrum? And one of the components is epigenin. So this contains a xanthone type of molecule and a flavone type of molecule. And also you can see a number of other peaks were also identified, and they are found to be epicatechin and kind of quercetin or hesperidin type of molecules. So this contains a rich diversity of flavonoids as well as tannins. And of course, this mangiferin is unique in this extract. So this, we also did the LC-MS spectra analysis of this anupana, and we could get a large number of these peaks and corresponding molecular weights. And from this, we could conclude that it contains these kinds of molecules in the extract, although to a smaller extent, like protocatechic acid. The major interesting ones are this epicatechin, epigallocatechin, and the flavonoids here, and hesperidin is a very important molecule in this, and quercetin, and morin, diosmin, and naringenin. So these are some of the flavonoids present in this one, and catechin is also present. And to Dr. Vijayalakshmi's interest, I would say that this also contains some kind of a polypeptides, a small peptide molecules which are all supposed to be cyclic peptides. 5 or 6 membered cyclic peptides are present in this one. And later I found that these small peptides are bacteriocins. They just arrest the bacterial growth in any medium. So that is a very interesting thing here, that our product contains, apart from this tannins and polyphenols, it also contains small polypeptides which are useful in controlling the microbes. So with that, we went in for kind of bioinformatics with the standard drugs and compare it with what are the components that we have got. And then we chose these 10 molecules here. And we went through the various targets, the NSP1, and you can see that hesperidine, we see here, this is the bar chart, which shows that it is the inhibitor of NSP1 with a docking score. And these are all the ways they are docking with the protein. Now you can see that embeline, this is embeline, which is embeline 2, which is docking with this NSP1, and B is hesperidine. So very interesting molecule. And compared to the standard drug ribavirin, and you can see that this ribavirin is higher in score, and followed by hesperidine and embeline. So this way you can see that two of the natural product molecules have got almost equal to the synthetic drug. And similarly in NSP2, the other target, you can see here the orbital is major, and then followed by the 1-phenylethanediol and then artemisinin and things like that. And here hesperidine is not that effective in this control of this peptide. This protein. This NSP4, and here again hesperidine is scoring very high compared to withanolide. Withania somnifera ashwagandha is being used now, and that is one of the important herbs for the control of these viral infections. And you can see here epicatechin has come into the picture here, and epicatechin is also a very good target molecule, a binder molecule for this target protein. And now you have this proteinase CL3CL4, which is a very important target. And here you find that epicatechin is scoring very well. And then comes the embeline and curcumin and so many other components. Aloin is also there. And we go to the next one, the NSP6 target. Here again, ribavirin is maximum and epicatechin. So like that, so I would like to go fast on these various targets, and instead of repeating the same thing again and again, you see that you can have this herbal ingredients doing much better than the synthetic drugs. And interestingly, this morayanin is a product of curry leaf. So curry leaf also, this is a carbazole alkaloid, and that also has got a very good role in this control. And as the Ayurvedic literature said earlier, they had a carbazole alkaloids and things like that. So here you see that the simple ones that we can get naturally every day is this curry leaf, and hesperidine is from lemon, and aloin, all those things are there, which are all coming to a very common herbs that are available in our marketplace, which can be used. Of course, you will have to go for making a good extract formulation and standardize them. So now this is the for helicase protein inhibition. Again, hesperidine, embeline are the major molecules, and exon. So I'm just skipping through all these things. So finally, to say that the product which we have prepared or which we have got already has got a number of molecules which show promise as an antiviral agent based on the bioinformatics study. And therefore, now we are in the process of trying to see whether we can take it forward with the government approval to screen it for clinically on various patients and see whether this can be of use. So the product is readily available. So that one adds to the gives an advantage of immediate application of this product. So now with the other products like the turmeric, you have this curcumin. These are the structures you have here. kind of a polyphenol and tannins. And this phonochiol is like a magneolol, which is a dimer product of carvone or carvion. So this dimer product can be obtained by using a dimerization reaction that has been enhanced by the lactase enzymes. So lactase enzymes cause a dimerization of the phenolic products. Now this lends support to one of the Ayurveda processes, which is called Asava process, in which the herbs are put inside and then a kind of fermenting agent or the microbial agent is added in the form of one of the other herbs like Dietary flower or something, and it is the container is closed for self-fermentation, and you can see that number of molecules can get converted. Although if you are going to do this on curcumin with the lactase enzyme, what will happen to curcumin? And whether that product will have much better effective efficacy in controlling the microbes. So, so now you take this ashwagandha, this is the product, and Safed Musli. All these are bioactive products, and Cactoliga orchitis, and this is Mucuna pruriens. Now, what we are trying to do is combine all these things and then combine it with the biotransformation process, where you have this the organic herbs or the compounds mixed and then reacted with an enzyme to see what kind of product we can get. Like you take a catechol and you get a bis catechol. This is the dimer you get, and you can identify the presence of this product here after transformation. And what is its effect on the microbial control? Is really this bis catechol is much better than the starting material. And this hydroquinone also undergoes transformation in this fashion, like a kind of coupling dehydration mechanism. It just couples with the water elimination. Now this kind of products have got their own efficacy in the bacterial or microbial control. This is another catechol. This 4-methyl catechol is also a product, byproduct from cashew nut shell oil, and therefore that will have a very good effect. Now, this beta-naphthol, and if you see one of those compounds which are controlling the virus in one of the targets, it contains a quinone or a kind of anthraquinone. So therefore, this embolin is also a quinone molecule, and this anthraquinone can do the can substitute embolin. And anthraquinone is obtained from aloin. But interestingly, dimer of this anthraquinone molecule is obtained from our senna. The sennocytes have got a dimeric product, dimer of this anthraquinone, not like this, but in a different way. That sennocyte is also a very good product which can be used wherever this kind of ketone is being applied. So therefore, we have in our arsenal a large number of these herbs that are useful for controlling this virus. This is menthol, and resveratrol is again another one. So I'm trying to see whether we can use the herb as such and also do some kind of this Ayurvedic transformations like Asava transformation and get the product, and that product can be used for evaluating the efficacy of our microbial control. So I'm cutting it short, and what I'm saying here is the herbs offer immense potential for identifying lead molecules for the treatment of infectious diseases. Like you can have this embolin type, and you can bring it to a very smaller type of molecules, even like a camphor type of thing, or caffeine. Even caffeine could be a very smaller molecule, can be very effective in some way. And then you can also be using this kind of smaller molecules and then subject them to a process called ASAVA using enzymes like lactase and then get a much better product. With this, I'm concluding my talk and maybe leaving the podium for answering questions. Thank you very much.
Thank you, Dr. Narasimhan. It's a very, very good talk. I have a question. I have the privilege of being the chairman, so I have the first question. So this hesperidine that you have tested,
yeah.
Do you know by any chance the target? Because hesperidine is already in use in blood thinning and blood related problems, because I have been using that as a patient long time ago. It was a combination of hesperidine and chymotrypsin that was given to me long time ago. So but that's a very, very interesting thing that you have found. The hesperidine is having a target. So did you find out a little bit more into that hesperidine pathway?
There is one correction, Vijay. It is hesperidine. That what you are using for blood thinner is different. Betaferin and that kind of product.
No. It was hesperidine. It was very clear because, and also I had a friend who is a medical biochemist and also medical from Harvard.
Okay.
And we were discussing about this hesperidine use, and then we had some work done on that. Unfortunately, is no more. So this do you have, because that why I am asking this question is, see, in some of these particularly COVID, the coagulopathy. So whether this hesperidine is interfering in that blood coagulation, which may be very, very interesting.
Your point is correct. This is one of the things which people are also worried about, using this kind of product, for whether it will interfere with the blood thinning, other molecules. And
no, it's important. Blood thinning is important in COVID control.
Yes, that is true. Yeah. So I think we have not done much work on the other aspects, but we have only found that in our product, this hesperidine is present. So that is a very interesting coincidence. And so this could give good relief for this COVID.
Because hesperidine is mainly in the inside of the citrus fruits, not outside, not the fruit, but in between that white part that is mainly rich in hesperidine.
Right.
So what is the source of hesperidine in your preparation?
Right now we are using that Unkarandi.
What is it?
Unkarandi.
Okay.
That has got this product and also mangiferin.
Okay. Oh, that's very interesting. Mangiferin is mainly in the mango seed, so this is very interesting.
Yeah.
Okay. Yeah.
Not of that family, but a different one, and still contains the mangiferin.
Maybe in isoform. Yeah.
Right.
Okay. I think I'll now give it to the podium for questions. I'm sure a lot of young people are having a lot of questions.
Sure.
I will request Dr. Priyankar Sen to take the 1 or 2 questions.
Yeah.
Okay. Thank you, Dr. Sanjit Kumar. Dr. Narasimhan, there are few questions.
Yeah.
Can Ayurvedic products be patented?
I have already filed a patent.
Okay. The next
This
question
is not exactly Ayurvedic. This is different.
This is not Ayurvedic.
It is not Ayurvedic. It is herbal product. Yeah.
Okay. The question is actually the Ayurvedic product can be patented or not?
No, Ayurvedic product as
No,
classical literature preparations, you cannot. But if you are going for a proprietary product based on herbs or any extracts or processes, that you can.
Okay. The second question is that whether Ayurvedic product or herbal product is permissible to taken along with allopathic drugs.
Yes, you can.
Yeah, of course.
That is why we call it.
Are there any pros and cons of it?
No, you will have to check the interactions, but you see, as such, the Anupana concept is mainly meant for that. So it is used for boosting the efficacy of the drugs. In our case, it is enhancing the efficacy of metformin.
Okay. The third question is that whether any herbal drug has role in cytokine storm, against the cytokine storm, which is a matter of discussion in COVID-19.
If you
mentioned, yeah.
can tell me what is the molecule that is responsible for the cytokine control or management, I can tell you which herb can be used.
Okay, I mean, there are ways to do it.
It is because see, the herbs are the arsenal of all types of molecules.
Yeah,
So any kind of analogs.
Okay. One attendee has asked that in that sanitizers, which are now well everywhere, why not any antimicrobial agent is used along with the sanitizer?
Herbal.
I'm not sure why they are not, but why should they not? They can use. What is the question actually?
That antimicrobial agent,
Yes.
why it should not be present in the sanitizers?
In the sanitizers?
Yeah.
Or maybe the cost.
Hand
Otherwise,
sanitizers.
That's what I said, cost.
Maybe the cost.
Yeah, but orange peel is already used.
Yeah,
but
see,
otherwise it's a cost, yeah.
Is the kill that you should get within that time frame. So alcohol kills within 2-3 minutes. And if you have any kind of material which can disinfect in a short time, so that's what you want, that can be used. And now actually we are planning to have some kind of a filter, the mask filter, wherein this will disinfect the virus rather than filter the virus. Because if you filter the virus, the mask becomes a carrier. They discard and it becomes a carrier again. So it is not good. So therefore, you need to disinfect the mask. So for that, we are now trying to have a filter solution, which you can spray so that will disinfect. So that, as you said, the antimicrobials from the plants can be used. The only thing is you will have to check whether it is as effective as the alcohol. We would like to go for a non-alcoholic sanitizers.
Okay, Sabris will go for next talk.
Yeah.
Okay, thank you, Dr. Narasimhan.
Okay, thank you, Dr. Narasimhan. Again, thanks very much for this very interesting presentation regarding what are the things that we are using regularly or some of the herbs, and then what are the constituents. Thank you very much.
Thank you, Vijay. Thank you, Sabresh. Thank you, sir.
Yeah.
Vijay ma'am, I will request you to introduce next
yeah, but you have to stop sharing Narasimhan's presentation and then put the profile of
Dr. Narasimhan sir. Please unshare your Dr. Narasimhan sir. Please unshare your PowerPoint file.
Yeah. Now we are ready.
Yes, ma'am.
Yeah. So, are we ready? Okay. So,
yes,
Dr. Ramaswamy is one of the young persons of the day today to come and share his very unique experience. He has done his PhD from Bharatidasan University, after which he went to Korea for some time. And from Korea, I think he took a long way to go to China, Nankai University in Tianjin. I think that is the university I had the pleasure of going in very early ages, because that was probably at that time only in the communist regime, only university having English understanding and English capacity, and one of the top universities of China. Because in that time when I went, I was told it's one of the top universities, and it was very good. So we have the pleasure and opportunity. I do not know Dr. Ramaswamy, but I think we will know him more now, and then probably we'll collaborate. So Dr. Ramaswamy had the unique opportunity of working and understanding the Chinese medicine. So some of the Chinese medicines are unique. Only China and India had this tradition of the natural medicine, as we can call it, from different sources, animal as well as plant, etc., and able to really cure and take care of many disorders. Either it is infectious, non-infectious, neuronal, everything. Because with my lot of experience with China, I was really impressed with that. And Ramaswamy has got lot of awards, and also he had good funding from different agencies. So I think we are very eager to listen to you, Dr. Ramaswamy.
Thank you, ma'am. Thank you very much for your wonderful introduction about me. Actually, I am the PhD scholar of Dr. K. Tamarachelvi of Bharadass University. You might know her.
Apadiya? Oh, then you are in the family.
Yes, yes, yes. So,
In fact, yesterday I was thinking, all these marriages and people go to Korea because Shankar went to Korea.
yes, yes, exactly.
Yeah. So I was thinking of that yesterday. So I didn't know that you been with Tamarai.
Yes,
Good.
ma'am.
Everybody knows Tamarai because she was the one who was instrumental in setting up all the structure aspects of my center. So
Yes,
thanks again. So we are getting.
She spoke a lot about you when I was doing my PhD with her. Okay, so will you, can somebody make me as a presenter because I am unable to share my screen? Okay, yeah. Now I could. Okay. Is my slide is visible to everyone?
No, it's not clear. It's not clear, Rajesh.
It's not clear yet?
Yeah, no, it's not clear.
Okay, I shared my slide. It's not clear yet?
No, not yet.
No, it looks distorted.
Yeah.
Are you having a Mac?
Yeah, I'm with Mac, yeah.
Fine, maybe the settings.
How now? Is it fine?
Problem?
No,
it's not clear still.
What should I do now? Should I connect?
Screen settings.
There must be some different options, right? Like in the Zoom.
What you do is you send your PPT by mail to Sabrish immediately by email. Then they will project it. Yeah, that's what we did yesterday for someone. Okay. Just send it to one of those mails. Sabrish.
Yeah.
Unga mail kodunga odane.
Yeah, I'll do it now.
I think he is having my email ID, ma'am. So
okay,
I think
then send it to Aisha.
Yeah, please do send.
Yeah, you can send it to Aisha.
All right.
Aisha?
Yeah, ma'am.
You project it for him.
Yeah, sure, ma'am. Yeah, let him send. That's what I'm waiting.
Yeah.
Did you send Dr. Rajesh?
Yeah, I'm doing, ma'am. Just a moment.
Yeah.
Sorry, because I don't know why it was not showing up, but I could do it. I could share my screen, but I don't know why it's not visible to you.
In between, I am launching the poll. Okay. Sabrish?
Yeah, tell me. Yeah.
I'm launching the poll in between. Okay.
Yeah.
Yeah, that will be good.
So the attendees can now answer the poll.
Did you send Dr. Rajesh?
Yes, ma'am. Yes, ma'am. It's like 60 slides, so I guess it's uploading still. Just a moment. Please refresh your email and please check, ma'am. Like, I think it will reach you.
No, still I did not receive Dr. Rajesh mail from you.
Yes, ma'am. Yes, ma'am. Yes, ma'am. It's showing is uploading.
Oh.
Uploading attachment.
What is your file size of the PowerPoint file?
Must be.
Otherwise, try to do once again sharing, Dr. Rajesh. Maybe it will work out.
Yeah. All right. Yeah.
The alternate, you can try it.
Okay.
Go down to the bottom at the green button. Yeah.
If it will be more than 25 MB, then you can share through Google Drive.
I mean, now can you see my slide?
It says opening.
Yes.
Full view. Full
Got
view.
it.
Yes. Now
All
we can.
right.
Put it full screen.
Full view. Full view.
Yeah. Maybe that it takes time to complete broadcast, because I do have a very poor network. I'm in a small town called Shankaran Kovil. So here I don't have like speed of internet facility. All right. Okay. So yeah, thank you very much to everyone. So those who are joining this. Yes, ma'am. COVID-19 disease control opportunities and challenges for drug discovery development, drug repurposing and alternative medicine, which is hosted by Center for Bioseparation Technology, VAT. So I'm very happy to be a part of this meeting, and I'm happy to deliver my talk about coronaviruses and traditional Chinese medicine. And this is going to be my 12th presentation during lockdown, and I organized like other meetings in my own Art of Giving chat with us, so which I will tell you later. Okay. So with that, I would like to give my sincere thanks to Vice Chancellor, Dr. G.B. Swanan sir, and the known person, Dr. M.A. Vijayalakshmi, former director of CBST, and Dr. Aisha Noor, who's given me this wonderful opportunity, and other organization committee members as well. Okay, so here goes my research history. Okay, so after my master's from Bishop College, I did my postgraduate diploma in bioinformatics from IISc Government of India. Later, I joined. Before doing my master's, postgraduate diploma in bioinformatics, I just been to Central Drug Research Institute, CSIR Lucknow. So where I did my master dissertation work. Then I did my postgraduate diploma in bioinformatics, and I joined AstraZeneca Pharmaceuticals R&D. I was working as a computer drug designer, taking lead components for microtuberculosis, that is an R37. And then I got an opportunity for doing my PhD from Bharatdasan University. And part of my work, I did it from Niri, Nagpur, under Hemant Purohit, and Natya Kaplamem, who are known person in metagenomics research. And then I moved to the Q's ranking top 100, Jeonbuk National University, which is in South Korea. Then my hungry for joining top 500, 100 university, it was not just like that. So I tried to find top 500 universities. Then I found Nankei University, as a Vijayalakshmi ma'am told about Nankei University as a premier institute, because the first Prime Minister of China, he was the alumni of this university. So it is considered as a premier institute in China. And then I was looking for top 100 universities. Then I found Zhejiang University, where I got my another opportunity for doing my postdoc. So now I'm associated with Zhejiang University, which is in Hangzhou, which is the Alibaba's headquarters located. All right, so I do have a charitable trust. What I'm doing, I am helping poor needy people in India. Also, recently I organized one international conference at Puducherry, where Puducherry CM and former Puducherry CM and the Tamil Nadu Industry Minister were present. And during this lockdown, I initiated world's first coronavirus discussion series. So far, I hosted more than 20 webinars. And then I organized one international virtual conference on the emerging infectious zoonosis with Chaudhary Charan Singh University, which is located in M.U.R. And right now, I'm organizing various international faculty research development for Indian researchers and faculties to overcome the difficulties during their teaching or during their research. All right, so I'm the founder of Voice, for that which my organization myself is World Environment Day Hero. Okay, so let's start with today's presentation. Okay. So I like the persons like Thiruvalluvar. Hope everybody knows. And recently, you could see like most of the politicians, even the Prime Minister, used to tell Thirukural to even recently at the border. Actually, like he spoke something to the soldiers. Okay, so because like he's a well-known person, and his holy book is like considered as a Bible for whoever wants to have a peaceful or happy life. So from his Thirukural, actually, I would like to give a quote of the chapter called Medicine. And நோய் நாடி நோய் முதல் நாடி அது தணிக்கும் வாய் நாடி வாய்ப்பு செயல் நோய் இன்னதென்று ஆராய்ந்து நோயின் காரணம் ஆராய்ந்து அறிவிக்கும் வழியையும் ஆராய்ந்து உடலுக்கு பொருந்தும்படியாக செய்ய வேண்டும். So since it is like two lines, so in English it's called as a couplet. Okay, so couplet is like two lines. Okay, so in English the couplet means disease its cause may be abate the ill. Let leech examine these, then use his skill. Okay, so that means let the physician inquire into the nature of the disease, its cause, and its method of cure, and treat it faithfully according to the medical book of rule. All right. So this is my disclaimer. Okay. So what are the materials that I used in my presentations, not mine. Okay. So all those information I collected from research or some open sources materials. And as an academic professional activity, today's presentation is purely voluntary involvement for my time, efforts. I will not receive any compensation such as commissions, honorarium, or remuneration gifts and vouchers from the organizers. All right. This is a wonderful word from George Santayana. Those who cannot remember the past are condemned to repeat it. This is what's happening with us. Okay. So we already have lots of issues in the past, so SARS, MERS, okay, but still we don't have any proper medicine or any kind of vaccine for it. Okay. Now we are struggling, scientists are struggling, and they are coming out with some vaccines, but however we are not sure whether it's going to support our lives. Okay, so let's see so according to Central Disease Control, 75% of emerging infections come from animals. Okay, and mostly from the wild animals, not from the pets or the domestic animals which are surrounded with us. Some animals can carry virus, but it will not get sick because it might likely to have the immunity for that particular infection or the virus. Okay, but what happens when the virus change from that animal to another animal as a host? Then that virus is going to be unpredictable and it's going to be very, very dangerous, and we are witnessing it. Okay, so when new virus emerges, okay, and people might not have the immunity, and in times, so what happens? Our body generates antibodies for the new virus, and so sometimes we are able to survive with that virus. Okay, what is so we know the SARS, okay, is also an infection caused by the coronavirus, and it is a zoonotic origin caused by severe acute respiratory syndrome, that is coronavirus, SARS coronavirus, or SARS coronavirus 1, or now we do have SARS coronavirus 2. Okay, so we witnessed the first pandemic of 21st century. This is a SARS coronavirus 2. The first identified strain of the SARS coronavirus species, severe acute respiratory syndrome, related to coronavirus. Related to coronavirus, okay, like SARS or coronavirus. The symptom caused the 2002-2004 the outbreak, okay, and it was first reported in the Asia, February 2003, and by CDC. And over the next few months, okay, what happened? The virus, okay, spread into 26 countries and 5 continents. So here are the events of how the SARS coronavirus spread. Okay, so in November 2002, okay, from Guangdong province, okay, so it was outbreak, and from then actually it in another 1 year, okay, from November to September 2003. So almost 1 year, okay. So it spread and it was uncontrollable. However, so it didn't make lots of mortality. So how it happens is contagious, as we know, right? The SARS coronavirus 2 has given lots of lessons, how the virus is spreading to one person to another person. Okay, so here's the whole story of SARS coronavirus 1. A person from Gong province, okay, got an infection, and he was accommodated in a hotel, Hotel Yam at Hong Kong. Okay, from there, okay, lots of the inmates of the hotel, okay, the guests of the hotel, got the infection directly or indirectly, okay, and then they were spreading to other people. Likewise, that spread to different parts of the world. Okay, here is the whole story of SARS coronavirus 1. Okay, next one is the MERS. MERS is called as the camel flu, okay, and this is a Middle East respiratory syndrome, and it is also believed to originate from bats, okay. However, humans are infected from camels, either during direct or indirect way. This spread, humans typically requires close contact with the infected person, okay, similarly to, but what about the SARS, what happens? So even you're not close contact with somebody, if the infected person coughs or keeping, or somewhere the person being actually from that actually get infection, okay. The first identified case occurred in 2012 in Saudi Arabia, and most cases have occurred in the Arabian Peninsula. However, that larger outbreaks happened in South Korea in the year 2015, and again in the Saudi Arabia in 2018. So here's the whole story, okay. The Riyadh case, November 2012, okay. So it started, okay. From then, actually, the virus has been spreading to various parts of the world. Okay, so here's a small comparison of SARS coronavirus and MERS coronavirus and SARS coronavirus. Okay, the data first detection was in the SARS coronavirus was 2002 November, and MERS was June 2012, and the SARS coronavirus was December 2019. The first location is China, and this one MERS coronavirus Jeddah, whereas the SARS coronavirus 2 is from Wuhan city, Hubei province. And epidemiological outbreak. Okay, so this SARS coronavirus spread mostly in the South China, Canada, and some other Asian countries. Whereas MERS, distributed into 27 countries, Middle East, Europe, Africa, Asia, and North American countries. Okay, whereas SARS coronavirus has been reported so far in 20 countries, Japan, Singapore, Thailand, and again, so we are ranking number 3 now in the SARS coronavirus 2 outbreak. So when it outbreak, okay, so during the season of winter, and where MERS outbreak happened in the breeding season of camels. Okay, so mostly China has affected this kind of coronavirus during winter. Maybe possibly that time humans going to be susceptible. However, we are surrounded with lots of viruses. Okay, we do have good viruses. So so it will be looking for opportunities when you are turning something weaker, or even system is going something bad, or the virus is changing from transferring from the animal to some other host. Okay, when it changes to another host, as I mentioned before, so the virus is going to be unpredictable and make lots of issues. Okay, so the host here, they said the virus must be from Rhinoplus sinicus, and the virus MERS, so far we didn't know either. And similarly, SARS coronavirus 2, we still don't know, but the Jain level studies are showing it must be from bat, but however, we don't have an actual clue how it arrived. And the intermediate host, okay, from bat, it transit to civet. It's kind of a wild mammal, okay. And similarly, they are saying from the bat, the host is camels. Again, here also it's a big question mark. We still know whether it's from man is Benedicta, but still so it's going, lots of research are still keep on going, and data is accumulating. We don't have proper information about the intermediate host of the SARS coronavirus 2. Okay, so here's the whole story. Okay, December 31, 2019, China informs the World Health Organization of patient with Mr. pneumonia. And January 1, actually, Huna seafood market was shut down. On January 7th, I took my flight from China, from Hangzhou. I moved to India for the conference which I mentioned. Okay, so so same time, they found that they named the coronavirus. And January 11, the first COVID-19 death was happened. And yeah, let's come to Indian scenario. Okay, so the first case was reported in Kerala in January 30th. And then actually, like, so the things like what you're like, facing through, maybe you're reading through like, social networking, or like watching through newspapers, or reading through newspaper and watching TVs. Okay, what is the source of this SARS coronavirus? Okay, the coronavirus source is believed to be like, this palm civets. Okay, so it's a wild animal. Okay, so this is the intermediate host which carries the virus from this civet palm civets to humans. Similarly, like, the coronavirus, okay, the intermediate host is believed to be the camels, okay, from camels who are like closely associated with the camels, okay, they get infection. And from then, the community spread, okay, the community spread actually, it happens when one person, like, moving around, or from that, actually, it will like spread to other people. Okay, so first the coronavirus identified in the year 1960s, and it infects like various humans. And yeah, so first the coronavirus was identified from a chicken that infected with the bronchitis virus, okay? And the two viruses from the nasal cavities of human patient with common cold that were subsequently named human coronavirus 229E and the human coronavirus OC43. Okay, so there are like other members of SARS coronaviruses are there, okay? That is like SARS coronavirus 2003, human coronavirus NL63, 2004, Hong Kong University 1 in 2005, and MERS coronavirus in 2012. And we are like having SARS coronavirus 2, that is novel coronavirus 2019. So most of these have been like an more serious respiratory tract infection. Okay, so mostly it infects our lungs. Okay, since 2000, there are like two coronavirus infecting animals has been all cause outbreaks in human, that is SARS coronavirus 2 and MERS, and now we do have SARS coronavirus 2, that is the disease which spread is called like COVID-19. In 2003, SARS coronavirus affected 8,096 people, which caused like severe pulmonary like pneumonia infection, and there were like 774 deaths, and the case fatality is 10%. And if as I mentioned before, like bats were like likely origin of the virus, which spread further. Himalayan palm civets, this one you can see, this is Himalayan palm civets, and Chinese ferret badgers, this one, and raccoon dogs. So these are like wild dogs, which are mostly like they sell in the food markets or like meat markets in China. Okay, MERS coronavirus 2, MERS coronavirus was identified in 2012, Saudi Arabia. Okay, since then the majority of human cases, as I mentioned before, like it went from human to human transmission. Okay, and obviously, like so it has the dromedary camels. It's going to be the important reservoir of this virus, and from then it infected many people, and the mortality rate was like 35%. Okay, so what is coronavirus? Is a kind of virus. Okay, it causes disease in mammals and birds. Okay, and it's common like symptoms are like you'll be having nasal congestion or a kind of like breathing trouble and sinuses, and also like it infects the upper. Okay. Most coronaviruses are not dangerous. Okay, I'm telling again. Okay, most coronaviruses are not dangerous, and we are like surrounded with lots of viruses, as I mentioned earlier. Okay, so when it like when it changes from the host to another, when it chooses the new host, probably the time it gets become the big issues. Okay, so the name coronavirus is derived from the Latin word corona, which means crown, a hollow appearance of the virus particles, as the virions, that have a fringe resistment of like a royal crown or the solar corona. You can see here, like this is a TEM image of electron microscope image of the coronavirus. So where you can see the spike kind of like appearance, which the scientists say this is like a crown-like appearance, and they named coronavirus because of the spike, like the appearance of this appearance. Coronavirus, viruses comes under the family like a subfamily of orthocoronavirinae and the family of like coronaviridae, and the orders comes under the nidovirales. Coronavirus like envelope viruses with a positive sense single standard RNA genome with a nucleocapsid of helical symmetry. And usually the coronavirus, like the genome ranges from 26 to 32 kilobases. Okay, here are the 7 common types of viruses identified by Center for Disease Control, Atlanta, USA. Okay, the common human viruses are like 229E alpha coronavirus, NL63 alpha coronavirus, OC43 beta coronavirus, HKU1 like beta coronavirus. Whereas these are like the pandemic or like epidemic viruses. Okay, there is MERS coronavirus, SARS coronavirus, and SARS coronavirus 2. So the same information like which I mentioned, and this virus has like 5 structural proteins. So one is the nucleocapsid protein, that is N, and envelope protein E, and the membrane glycoprotein M, and spike glycoprotein S. So yeah, so as I mentioned before, like spike envelope membrane and the nucleocapsids are like 4 important main structure of these proteins. Okay, and yeah, so the receptor like it uses like an angiotensin like converting enzyme 2 ACE2. Okay, and so same for like SARS coronavirus 1 and SARS coronavirus 2. Okay, both are using the same cellular receptor, and some coronavirus like has a shorter like spike-like protein called hemagglutinin esterase (HE). Whereas like MERS has a DPP4 receptor. Okay, so I will show you like on my next slide. Okay, this is again like the virus receptor. Okay, so these are the natural hosts. I mean, like it's believed the virus is from the natural host, and whereas the intermediate host is believed for other viruses like cow, bovine, and and the civets and the camels. Okay, so these are the viral receptor. Okay, mostly like the SARS coronavirus has the ACE2 receptor, whereas the MERS has like a DPP4 receptor. And this is like SARS coronavirus 2. Okay, the genome size, as I mentioned before, the length of nucleotides is like almost like 29,000 to 30,000. It is believed to be like for both like SARS coronavirus 1 and SARS coronavirus 2. Okay, so it has like 11 open reading frame for both SARS coronavirus 2, SARS coronavirus 1 and 2, and MERS coronavirus 2, and it has like 4 structural proteins, and both like for the SARS coronavirus 1 and 2, and also for MERS coronavirus. And yeah, the spike protein has like more than 1,255 amino acids, and whereas like MERS has like 1,353, and it has like S1 and S2 subunits, and it has like non-structural protein. There's like at least 5. It is present in SARS coronavirus, and whereas like MERS has like 16, and it has like other accessory proteins like 8 accessory proteins, and for MERS has 5, and also it has like gene order of like 5 replicates or one AB spike, and there's like the other 4 that are structural proteins. Here is like a genome of like SARS coronavirus 1, and you can consider the SARS coronavirus 2. Whether it has like only just 2 kb of difference of like genome size. Okay, so this is just a comparison of both the genome of SARS and SARS coronavirus and MERS coronavirus. So again, the same one, like WIR of 1A, WIR of 1B, and the spike protein, and you can see that envelope protein, membrane protein, and nucleocapsid. Okay, so these are some of the basic information of the protein, and you can see like S1 unit, subunit, and S2 subunit, and the size of the genomes. Okay, here is the life cycle of the SARS and MERS. Okay, so as I mentioned earlier, okay, SARS coronavirus virus uses a receptor ACE2, whereas MERS uses receptor DPP4. Okay, so through that it enters, and the rest of the story. So it goes and multiplies, and the replication happens and transcription happens. Okay, from then actually like it multiplies inside our body. So these are the common symptoms. You like know all those things. Okay, the SARS MERS have an incubation period of 5 days, and yeah, so the common symptoms are like fever, chills, coughing, malaise, myalgia, a headache, and yeah, so some people are having like diarrhea, vomiting, nausea. Okay, so these are some of the like common symptoms of SARS and MERS. Okay, and MERS has something like different. Okay, so MERS like infects the kidney. Okay, and not only lungs, also like it infects the kidney. Okay, includes diabetes mellitus, hypertension, cancer, renal, and lung disease, and also other co-infections. Okay, here you can see like in the comparison of like common symptoms of SARS, MERS, and COVID-19. Okay, so hyperproduction more than 38 degrees Celsius will be having, the infected person will be having, whereas MERS like having fever, cough, polypnea, and similarly like the fever, cough, and fatigue. So almost the symptoms are very similar. Okay. Okay, these are some of the clinical characteristics. Okay, let's move to the treatment. Okay, Okay, so far, like, they are having, for SARS and MERS, there are no medicine, okay, and there's no treatment at all, okay. There's no drug which can cure the earlier one, as I mentioned with the Santayana's word, okay. So we don't have like any kind of like, we didn't learn anything, or we didn't like, make any kind of drug or vaccine for SARS. So if we would have like done something earlier, so it would have like, supported our lives at this moment, but we don't have any kind of like, a drug or any kind of vaccine which can like, cure the SARS coronavirus infection. Okay, here there is a treatment method, okay, host protease inhibitors, which was effective on mouse models, okay. But however, like, so these are only in the level of like, animal model studies, okay. The common feature, none of these therapeutic agents are approved for commercial use in humans, okay. So far, like, you can see ribavirin and lopinavir and ritonavir, okay. So they use different kind of interferons as well, okay. But and also they used plasma therapy, okay, called plasma therapy. But still, like, still actually it was not like, support the lives of humans. Okay, vaccines, okay, similarly vaccines, okay, they use inactivated virus vaccines, live virus vaccines, okay, viral vector vaccines, subunit vaccines, DNA vaccines. So all of them are under trial, okay, but no, so far no like, vaccine has been done, even for like, from the past, okay, from SARS as well as for MERS. Okay, so let's go into the core idea of like, traditional Chinese medicines, okay. So I'm not going to like, give like, so how they are making all those things. So I'm going to share you like, what are the, how like, China like overcome by using traditional Chinese medicine so far, okay, how they manage, and how the like, patient recovered. Why India like, still we are like, having issues with like, using Siddha or Ayurveda, okay. So what are the like, differences, okay, why so we do have like such issues. So with that issue, like, so I'm going to give a brief idea about like, what are the traditional Chinese medicines so far used during this I mean, like, any kind of viral infection which is happening in China. Okay, so conventionally, like, so what are the like, so the Western medicines, what we are using, all these, okay. So, but this is not like going to like, support us. This is just a kind of like, life saving, or like at the, I mean, like a point of like, giving, I mean, like, providing like drugs, okay. It's also like, depends upon the patient health, and obviously they will see the physique and their age, okay. So everything will be considered before like, given any kind of like, these kind of drugs provided. Okay, let's see what is traditional Chinese medicine, okay. It's a very oldest like, practice of medicine in Asian countries, especially in China, Japan, Korea. So it's, I mean, it's believed it's like, more than 2,000 years old, okay. And yeah, so the treatment is based upon the clinical use and wide-ranging effects in the treatment, and of like, various diseases, and especially like, chronic diseases. And yeah, the first, I mean, like, the Nobel Prize winner of like, physiology of medicine, like Yu Tu, and his discovery on like, artemisinin, this is a key anti-malaria drug created with the same millions of life, that give us some idea about what is like, traditional Chinese medicine, and people like, started like, practicing not only in China, even like, other part of like the world. Actually, they started using, and at least they come to know so what is the traditional Chinese medicine. Okay, so globally, scientists are frequently looking for like therapeutic agents from traditional Chinese medicine, which is a rich source of potential leads for drug development. Similarly, we also have such kind of like, like phytochemical, like plants or herbs, but still like we need to do proper formulation, and it should be, properly done with clinical trials. Then it can be like, used for humans. Shifting the current drug discovery tide from finding new drugs, okay, screening natural products may be helpful for overcoming more investment, fewer drug challenge. Exactly. So this how like, so they are like, the current drug discovery system is working, okay. Because like, so we do have lots and lots of drugs, okay. So, and we are like investing like lots of money, but we do have like, very few drugs. So we should like, go for something more reliable, and also which supports our lives. That's why the World Health Organization estimates more than 80% of the world's population relies on herbs for their primary healthcare needs. Even like you can come to southern part of Tamil Nadu, especially like Tirunelveli districts, you can go anywhere. They used to give suku kapi. So in the morning, like 4 a.m., like anytime in the evening, you can go, you can find the people used to have suku kapi, because suku kapi has the ashwagandha, okay. So which is a very like, kind of like the herbal, which supports our lungs, which is not allowing any kind of like, viral or bacterial to get infected. Okay. So that kind of like, primary health care, we are like, taking care, especially in Tamil Nadu, I could say. I'm not sure about like other parts of India, probably they might have like different kind of practice. So I'm just sharing like, what I know. Okay. So here's the Chinese traditional medicine style. The Yellow Emperor inner canon, okay, that is this opinion, okay, Huangdi Neijing has been the most influential treatise for more than 2,000 years, okay, and they have like treated plenty of people, okay, so those who have been like being a soldier in the wars, okay, so they learned like all kind of like practices after that, okay. So they use 5 phases of concepts of nature, that is wood, fire, earth, metal, and water. You can see here. So they use these 5 key as a drug development in traditional Chinese medicine. Okay, so you can see like water. So they use they mix with salty and kidney. Okay, and for like kidneys is cinnamon and wood. Okay, the wood they consider sour and should be like it will be taken care by the liver. Okay, and heart, for heart they give like bitter kind of stuffs. Okay, and for spleen they give like sweet kind of like things. Okay. And yeah, the astragalu like acrylic, okay, metal compounds actually like given for like treating lung kind of infection. This is like they are like 5 phases of like information. So for they recorded in their books, and they are using the same kind of things for like treating the infections. Okay. And yeah, similarly, actually, we Indians also have the similar kind of like treatment. I mean, like following method something. There's a 4 element thing. Okay, earth, water, fire, and air. That was like by the Buddha. Actually, like the Buddhists, they follow this kind of ideology. And still, like so the 5 phases also like used in like Greek, like European countries, they also follow the 5 phases. Okay, these 5 phases. I mean, because maybe like so we would have actually like from India, it would have been to China. We know the Bodhidharma actually. Even like if I speak to some of the Chinese, they used to tell me Dhamma Muni. Okay, Dhamma Muni means like the Bodhidharma actually, who was a Pallava king actually. I mean, you'd have seen the movie called Yela Marai. Okay, so you'd have been there actually, like he started like practicing something. The Chinese probably from that actually they would have also learned not only the martial arts, also they would have learned this kind of like the 4 elements. Probably like they added 5. The wood has been added into that actually. Probably they would have like learn this kind of system from us. And also the Greeks also, like the Europeans also learn the 5 phases of like treating the humans by using the traditional any kind of like traditional medicines. Okay, from place to place it changes. Okay. Okay, so here's the book name. Okay, so like we do like some Ayurveda and Siddha books, right? Like similarly, Chinese also having books for like for treating different kind of diseases, and it is noted like before Christ actually 1046 Zhou dynasty. Okay, Zhou Yi. So it's called like Yi Bing. Okay, Yi Bing is like common cold. I mean, like it thought like that Yi Bing was a different from common cold, but it was a disease actually leaded to epidemic. Okay, so from then actually like there have been like lots of like different kind of books actually they wrote, and also they are like following up, and you can like I am going to show you like in the next couple of slides the drug name also having this kind of like information over there. Okay, so what is the TCM? Okay, TCM therapeutic efficacy usually property of multiple herbs. Okay, they use multiple herbs and constituents. Okay, and they give it to they make as a formulation. Okay, proper formulation to improve the efficacy of multiple targets. Okay, so they don't like make something for like something some particular infection. Okay, so they make you mix multiple herbs and actually they formulate and properly like they do the formulation, then they give it to like multiple targets. Like it can be like any kind of viral infection. Okay, probably that is going to be the cure for that infection. Okay. Okay, so based on symptoms and patient's characteristics and guided by TCM theory. Okay, theory means the books. Okay, so the previous stories. Okay, theory means probably they would have consider as the books actually what they already like read or like already they practice. Okay, and also from the earlier formulae. Okay, they design like new kind of like crude drugs. Okay, and it can be like with a mix of like different kind of like ingredients, and it will give like maximum quality to cure from the infection. Okay, For improving the health, actually, practice advocated combinatory therapeutic strategy based on overall symptoms and symptoms of symptom, and often prescribed to formulate that works together harmonies to achieve ideally therapeutic efficacy. Yeah. So even assessing pharmacies, what when I was there, actually, what the scientists or those who are working there, what they're doing is they're going for combinatorial chemistry. I mean, most of the drugs what we are available in the market is it comes with combinatorial, two different compounds, they mix and they come because they want the efficiency to cure from the infection or from the disease. Okay. So similarly, actually, traditional Chinese medicine also comes with the combination of different kind of herbs. Okay. A single herb already contains thousands of compounds. Okay, so you can see here this is a single herb. Okay, it contains different could be thousands of compounds, whereas formally contains multiple herbs, possess multi-component, multi-target therapies. Okay, so it may have different kind of it can work with the different targets, but that should be properly formulated and should be taken by the proper clinical trials. Okay. Typically, formula which consists of several herbs would comprise hundreds of compounds as a complex system that contains lots of compounds with the diversity in it will be having different chemical structure and it will be having different biological activities and interaction among it may interact with each other compounds. Okay, and also it contains different kinds of components. Okay. However, so this should be properly, as I mentioned earlier, okay, so it should be properly formulated, okay, then only it can be given for any kind of patients to be treated. Okay. And yeah, so many Chinese therapies, okay, used in co-treatment but not mono treatment. So as I want to share something. So whenever I used to go to hospital, they don't only give the Western medicine. Okay, so they always admit Chinese traditional Chinese medicine along with the Western medicine. Okay, so that's how they practice in China. They don't in India actually we used to only admit with only the Western medicine or either Ayurvedic. We follow in this way, but whereas in China they mix together. Okay, so they use as a co-treatment but not mono treatment. Okay, and yeah, so because of mono treatment, it may take time. Also, it may have some other issues. When you go with the co-treatment, and it will have very less side effects. Okay, probably there is a reason actually they always give Chinese traditional Chinese medicine along with the Western medicine. Okay, so how so what is the principle of traditional Chinese medicine? Okay, so traditional Chinese medicine, okay, when combined with chemical drugs, it has a multi-component. Okay, so it is multi-target as I mentioned before. So it not only cure only one viral, it's not target based. Okay, so it's a multi-target. Whereas allopathy or Western medicine is target based, one particular gene or protein. Okay, so with that we develop medicine. Whereas the traditional Chinese medicine so it has some different kind of compounds and has a multi-target and also multi-pathway characters and poses unparalleled advantage when faced with miscellaneous diseases. So whatever these comes, they treat with this traditional Chinese medicine. Okay. Traditional Chinese medicine have these four formulae, these four elements. Okay, so the first one is the monarch, Jain, minister Chen, assistant Zhou, and servant Shi. What the monarch? Okay, these are four elements. They actually they segregate the elements in this way, the medicine in this way. Okay, and the monarch, okay, that aims at the cardinal pathological symptoms of disease. Like we play chess, right? We play chess. We used to have soldiers. We used to have elephant. We used to have different coins. Similarly, actually, they also play in this way. So first, the monarch aims at the cardinal pathological symptom of a disease, and minister what it is as his monarch to treat other secondary symptoms if applicable. And what assistant does, what assistant servant does, mainly coordinate the formulae and facilitate the performance of monarch, monarch and minister. Okay, so this assistant servant supports the both because this is going to be the more efficient. Okay, and decrease their side effects. Okay, this is like a chess game, so they actually distributed the preparation of traditional Chinese medicine in this formula. Okay, in this four elements method, and they work together harmoniously to achieve an ideal therapeutic outcome. Okay, so far 11,100 plant species and 1,581 animal species and 80 minerals.
Dr. Ramchand Rajesh, I think we have to.
Yeah. All right. Okay. So can I take another 5 or is it okay or what?
Yeah, quickly, because I think the others are waiting for the question and discussion.
All right.
If it's one room, it's easy, but because people are all around the world now, maybe from different places.
Yeah, I got it. Yeah. Okay. So I will show like what are the traditional Chinese medicine has been used. Okay. This one has been efficiently used, Qing Fei Pai Du Tang. So as I mentioned, okay, so this use glycyrrhizase radix, and this is the most efficient one. There's ashwagandha that we're using, okay, and these are the components that is used for Qing Fei Pai Du Tang, which they used for treating COVID-19 patients, and also they used a Xinhua Qinggan granule, okay. It was also used in 2009, okay, in influenza, and it used like 12 herbal components. And you can see the negative of the patient from coronavirus to negative in 2.5 days, okay. But sometimes it takes 8 days. And Lianhua Qingwen capsule, okay, that uses 13 herbal components. And Xuanfei Baidu granule uses 13 herbal components. And this injection, okay, Shu Beijing injection, okay, so it was also used in 2003. It uses 5 different herbal extracts, and it detoxify and remove blood statises from our body, and it also treats sepsis. And it was used in different hospitals, with combined. It also combined with different Western medicine. So far, 7-10 cases has been cured, okay. And yeah, the Huashu Baidu formula, okay, this was advised by China Academy of Chinese Medical Sciences. It uses 14 herbal components, okay. This detoxify, remove dampness, heat from body, and also it maintains the inner body balance, okay. And it it shortens the stay in the hospital. Also, it improves the lung condition. And yeah, lung cleansing and detox, okay. This medicine is more important, okay. So this has been given to more than 1,264 patients and different 10 provinces, okay. And out of 1,264 patients record, because this lung cleansing compound, okay. So Chang Han Za Binglun, okay. So it was invented in the year 2020 by Chang Chongqing, and this medicine clean our lungs, okay. The infected people Okay, these are some of the Chinese patent medicine, okay. Huaxiang Chengji capsule, Lianhua Qingwen capsule, Jinhua Qingwen capsule, Jufengjie capsule, Tan Reiching injection, Xianping injection, okay, Shu Beijing injection, Shifu injection, Chengmai injection, Angong Ni Huang pill, okay. These are these are approved, okay, by the Chinese government, okay. So this is Chinese FDA approved, okay, drugs which is used for coronavirus, okay, mostly antivirus, antipyretic, okay, anti-inflammation. Okay, these are some of the composition of this one, but I don't have time. I think so I thought actually it would be more than 1 hour. Okay, so what are maybe okay? So yeah, so even India do have lots of herbals, okay, that should be we should be properly discovered. Also, it should be we should restore our ancient literature and also the traditional way of treating infections, okay. And yeah, so we should have more plant-based biotic compounds that should be identified and formulated, and should be have proper clinical trials before giving to humans, okay. With that, actually I should conclude, okay. So my presentation. Okay, so I would like to share, if you have a quick 1 minute, okay. So I'm organizing one day, International Faculty Research Development Frog, supporting Assam Floods, how to become a successful researcher, which is going to happen on August 9th. And also, if somebody actually asks a question how to patent our formulations, or whatever might be actually how to do that. So for that, I'm organizing 2 days certificate course on intellectual property rights. And the speaker is former founder director of Center for Intellectual Property Rights, Anna University, Chennai. And if anybody interested, they can contact me. So if you have any questions, I'm happy to provide the answer.
Okay, thank you, Dr. Ramsamy Rajesh. So the interesting part of the Chinese practice, I think we'll have a lot of questions. Yes. I think also, as you mentioned, that monarch, minister, and servant. That's more or less the concept in every medicine with different names, because the formulation takes up the important thing. Because I know some of the pharma industries in India were asking me, Vijay, do you know any formulation person? That is where we are really lacking. So that servant part is the most important. That is what is formulation. Of course, yes. Chinese won't reveal all the details. I know them very well. But it was very good to know about how the Chinese medicine is being used in some of the things, because having had a lot of Chinese interaction, I knew some of their herbs, what they were using. Yeah. You know, they use chittarate, what we use also, same thing. So that's a very interesting thing. So thank you very much. I'm sure the audience have more questions.
Yes, please.
I will request Dr. Aisha Noor to ask the question.
Thank you, Dr. Sanjay. Yeah, to Dr. Rajesh, they were asked, one of the participants has asked that the usage of traditional medicines like Ayurvedic along with allopathic medicine. So how effective is the co-treatment in the case of Chinese treatment method?
Yeah, so as I mentioned before, okay, so when I used to go to hospital, mostly like what doctors in China does is they always like admit traditional Chinese medicine, because we use a kind of like anti-inflammatory or anti-analgesic or like antacids. That also a chemical compound, right? Whereas like in China, they give the natural way of like that's from the herbal. Similarly, we should also like practice such a kind of like admitting allopathy along with the Western medicine. Probably that is going to like support. We cannot like have any kind of side effect. Also, it will like prolong our stay. Okay, so that is more important. So that's why I'm advising. So we do have lots of like a similar kind of like Siddha Ayurvedic practice in India, but the problem is we are not admitting together. We are like giving it separately. So we are not seeing any kind of like kind of like benefit to the human lives.
Yeah, which is one more related to the same question they have asked. One more, the patients when they are being given with the combined medicines, is allopathic and traditional medicine. So how would we know that the health benefits are attributed to which medicine? Is it the modern medicine or the Chinese medicine or our Ayurvedic medicine? So how do we know that? So
no, that's what I'm saying. That should be done through proper clinical trials, right? So when you're like admitting all those things, you should like admit into like different kind of like animal models. From that you will understand which is having, see, the traditional Chinese medicine or supportive, okay, life support. It's not like some medicine will have some effect. Some medicine will like as I mentioned, like this have like 4 elements: servant, monarch, and that kind of like play will be there. Every element, every compound has that kind of play. So they know which should be like admitted along with the Western medicine. That's how they're like life.
You can see a simple example, like for example,
yes,
it is not exactly medicine, but when we are having fever, we take antibiotic, everything, and also we make kanji with dry ginger. That's a very similar thing. Yeah.
Yes, exactly. Yeah. So ginger. Yeah. As ma'am mentioned, okay, ashwagandha we can take. Or at my home also, we take paracetamol along with chuku kapi.
Yeah.
We have like cold season because my hometown is close to Tenkasi. Okay, so we used to have like cold weather during during January or December. So many people used to have some kind of flu. Okay, so what they does, they practice paracetamol along with the suku kapi. So probably we don't know like which one gives the remedy, but however we are getting treated. That is more important.
Yeah. So one more, I think I can ask the question, last question maybe. So how the bats or the other animals which are the carrier of this virus can sustain their lives with content of this coronavirus?
Sorry, can you come again? The question?
Yeah, hypothetical, like how the bats or the other animals which are the carriers of this virus can sustain, I mean to say they are alive with the content we're carrying with this virus. That is the question.
Yes. Yeah. Bat, I mean, somebody like in my my program actually, he mentioned bat carries like more than like 200 dangerous virus, but it will not give any kind of infection to us, okay, directly, because bat never give infection to us. Hope you understand, okay? When bat gives the virus to some intermediate host, then it gets a problem. We go to temples. In the temples, we could see lots of bats will be there, right? You can see any temples you go, like lots of bats are there, but bats never gives infection to us. But when the bat gives the virus to some intermediate host, then it becomes very dangerous, okay?
So is there any scientific, any viral study, clinical study was done with traditional Chinese medicine with regard to SARS-CoV-2?
Yeah, I mentioned, right? I have shown you, like, there are like many. They actually, like, I have given like more than 5-6 medicine, okay? There's no single medicine, okay? So it depends upon, like, we are having Siddha, Ayurvedic, okay? Similarly, yes, they see the patient condition, they see the patient age, and also they do the CT scan or like they do the X-ray of the lungs, because mostly Chinese traditional medicine, okay, what they target is they target the lungs and also liver, okay? This is the most important, because every drug should have ADME property: absorption, metabolism, excretion. Absorption, digestion, metabolism, and excretion. Okay. So similarly, the liver is more important for digestion, metabolism, right? And so that's why, like, the traditional Chinese medicine, what it helps, they want to improve the lungs. Similarly, they want to improve the liver. So I feel, okay, so what are the components they are providing? Actually, like I read the components of all the medicines, okay, traditional Chinese medicines. Mostly the target is lungs and the liver. So I think probably we should also, like, find the kind of, like, compounds. As ma'am mentioned, ginger supports our liver, right? All the toxic compounds are removed from the liver. That's why, like, we are having the ginger tea, or like soup kapi we are having. So improving the liver is more important. What are the drugs we are giving? Improving the liver is more important.
Thank you, Dr. Rajesh, for your wonderful talk. I think now Vijay ma'am will take it up now.
Thanks a lot, Rajesh. I think the most important thing is the way of life that the Chinese have, and integrating their medical treatment every day in their regular life. That is more important than just saying this is for this disease, etc. That is the traditional Chinese way. Yeah. Thanks a lot for your informative talk. And I think I'll give the mic to Professor Krishnan, who will probably conclude. Thank you.
Thank you, Professor Vijay. Thanks a lot for sharing.
Thank all the speakers this morning, which was a very good, excellent experience.
Exactly.
And I think we got a lot of information and comparison between the Western approach and our thing, and it's a very good morning. Thanks, everyone.
Yeah. So I take this opportunity.
You can unshare the screen, Dr. Rajesh.
Okay, ma'am.
Yeah.
Okay, ma'am. Okay, sure.
Yeah, thank you. Yeah, sure.
Okay, I take this opportunity to thank all the speakers, Dr. C. N. Ramchand, Dr. Ramesh Warrior, Dr. Narasimhan, Dr. Ramaswamy Rajesh Kumar, for wonderful lectures, very, very informative, extremely useful lectures, and I'm sure us and students, they really liked it. Thank you all for taking time off to come to this podium and present your work and explain a lot of things. And I take this opportunity to thank all our students, especially Neha Deora, Vijay Lakya, Spurti Babu, Miraculin Prasanna. They've all done background work. Faculty colleagues, all of them, they helped to run this conference. They invited speakers, Aisha Noor, Sabrish and Kishore, Sanjit Kumar. They've invited speakers, of course, Professor Vijay. And many of them have served as resource people. And I'd like to thank Professor Vijay from guiding us from the back. And I would like to take the opportunity to thank our management, our Chancellor, Dr. G. Vishwanathan, our Vice President, Mr. Shankar Vishwanathan, our Assistant Vice President, Ms. Kadambari Vishwanathan, and of course, our Center for Technical Services to host this event. Finally, importantly, all the participants, they come in large numbers. They came, patiently waiting for hearing everybody's lectures with a lot of enthusiasm. Without you, this could not have been successful. So we did 2 conferences, including this on COVID-19 disease control. One was focusing on vaccines, biotherapeutics and diagnostics. Second one was on drug discovery and development, repurposing and alternative medicine. So it gives a perspective, and I'm sure many of you will be excited to work in this field. And coronavirus, it will not disappear, although we may find a vaccine soon and medications, very good medications, etc. Till then, I would like to request all of you to stay safe. We had Dr. Ashok Ratan, he's a medical doctor who participated in the last event. He just sent me a message. Please request everybody to stay safe, follow all the physical distancing, wearing masks, etc. He says people should not take things for granted, because once they get affected, it takes a long time to recover, even for younger generation of people. So this is what his experience he's observing in Delhi. So he asked me to pass on this message. So I'm passing on to each one of you. Thank you all. Thanks for a wonderful event.
One
Big
second.
thanks to everybody. Yeah.
I have something, Krishnan.
Yeah, please.
Yeah, so I would like to thank Professor Krishnan for getting us such a good team. And also, I would like to take this opportunity to wish on behalf of every one of you, on my personal behalf, Neha, a happy birthday.
Oh, great.
One of the key people who made everything. So I would like to say happy birthday to Neha on behalf of everybody.
Happy birthday, Neha.
Happy birthday, Neha. Happy birthday, Neha.
Right, thank you. So we'll again try to get back like this in one, perhaps after some time. And till then, goodbye. So all good things should come to an end. We should go back to our work. And I think, sorry for the delay. You're all waiting for your lunch. Please have your lunch, relax, and then so we
Bon
start our
appetit.
work. Yeah, thank you. Thank you so much.
Thank you, Archana ma'am. Thank you, Vijay ma'am. Vijay ma'am, thank you.
Thank you, thank you, thank you.
Great.
Thank you, ma'am. Thank you, Krishnan sir. Thank you.
Thank you. Thank you. We'll meet sometime. Yeah.
Thank you.
One small announcement, sir. For participant, they will receive the certificate within 10 to 12 days. Not immediate. 10 to 12 days, they will receive the e-certificate.
Right.
We have to prepare name and institute. It will take time.
Right.
But if they don't give the feedback, they don't receive the certificate.
They have given the feedback.
They have
already.
given, ma'am. They have given.
They have done.
Thank you. Thank you.
Okay.
Thank you, sir. Thank you, ma'am.
Okay.
Thank you, Aisha. Bye.
Bye, ma'am. Bye, bye. Take care.
Bye, Kishore. So,
Bye,
Sanjeet, bye.
Bye,
So,
ma'am. Bye.
see you guys. See you guys.
See you.
I'm going to squeeze each one of you. So, be ready.
See you, Vijay. Thank you.
See you. Bye.
Okay, thank you, ma'am. See
I'm
you, ma'am.
ending the meeting.
Okay,
Yeah.
sure.
Okay. Okay.